Very good morning, Dr. Sekharappa sir. Sir, actually participants are join the webinars. So we will wait for next 5 minutes. Soon we will start the webinar. So kindly bear with us.
Okay, sir. Okay.
Thank you. Thank you, sir.
Hello.
Okay. Shall we proceed further, sir?
Yes, Honorable Dr. K.G. Patel, sir, Dean Faculty of Agriculture, is also here with us. And today's guest speaker is also here with us, Dr. Sekhrappa, sir. And all the dignitaries also here with one virtual platform of national webinar. So we can start.
Okay, okay. Thank you, sir. Good morning, all. I greet all of you, the delegates and district news participants present over here. And thank you for joining us on the fourth day of the webinar series that is on sustainable agriculture through natural resource management. It's my pleasure to walk you through the webinar, introduce you the experts and delegates that have come across the country and share their knowledge and expertise to boost up our mental strengths. Today's webinar already reached more than 120 participants from the across 20 state of the nations, and this webinar really a true impact event at national level. Before we start and proceed further, I am very much glad to introduce our today's dignitaries, Dr. K.G. Patel, sir, Dean Faculty of Agriculture, Navsari Agriculture University in Navsari. And I am also witnessed the gracious presence of Dr. Y.P. Singh, sir, Principal Scientist ICR, as well as Dr. D.D. Patel, sir, convener of the webinars. So I am very much eager to listen something from our dignitaries. So I invite Dr. K.G. Patel, sir, to say a few words as a opening remarks of today's webinar, sir.
Good morning, everybody. Today is the fourth day of our national webinar on sustainable agriculture through natural resource management. And really, the webinar organizer, Dr. D.D. Patel, co-organizer, Dr. Tushar Patel, and whole the team is doing very well job. Actually, after the inauguration of the seminar on very first day, Dr. Chaudhary Sir DDG was and Dr. J.D. Patel, our Vice Chancellor, was with us. So with the inauguration, today is the fourth day. Really, all these renowned speaker has given a nice talk, and around 200 participants have participated in the this webinar throughout these 3 days. And actually, it is interesting thing is that the as Tushar told that it is our great pleasure that around 20 countries participants are with us and they are enjoying the seminar because today the demand is fulfilled in this webinar because as we are talking about organic farming, better use of better resourcing, all the components are included in this webinar. So the participants are getting the latest information regarding this better resource management. And today, actually, there are 5 speakers. They are all the very renowned speakers. The first speaker is our Dr. Sekhrappa, insect pest management in organic farming through microinsecticide. It's a very interesting topic because everyone is going there. The most of the cost is there in our crop management is from plant protection. So with the organic insecticide, it is the minimum cost and the minimum hazard and with the environment. It is very good for our future generation. And second speaker, Dr. Lalit Mahatma, is also with volunteers of par excellence to manage natural resources in agriculture. His lecture is on micro. Number of the micros are there that are very important in agriculture, and they are very good natural resources to have useful for the plant protection. Similarly, role of entomopathogen in insect pest management will be given by Dr. Jetha. He is also a very good scientist and he has a depth knowledge in this topic with practical performance. So Dr. Jetha will also give very nice talk. That is the important thing. Nutritional intervention to mitigate methane production in ruminants. So this is also important thing because our animals are there and they are also very important thing. So Brishketu can give very nice talk. And enhancing water efficiency need of an hour, Dr. Ajat Patel. Everyone knows that our soil is day by day degrading because somewhere there is a deficiency of water, somewhere plenty of water. So farmers are using plenty of water in their agriculture. So water like problem are there. So how to manage the water in agriculture? That is also important thing because water is our natural resources. Water is somewhere limiting factor, somewhere is excess amount. So there are many things to reduce the pollution in the water and to use the efficient use of the water. That is also important thing. So many things will be narrated in this seminar, and this seminar will be useful. Today's talk are very important for this webinar. So again, I congratulate Dr. Deptt Patel and Dr. Navsari for today's speaker also, and I hope that all the participants will interact in a fruitful level, and this today's talk will also useful for the people also. So again, thank you very much to invite me to interact with the participants. Thank you very much.
Thank you so much for your appreciating words, sir. And thank you for introduce our today's speaker, sir. So without further ado, I request Dr. Deptt Patel, sir, to introduce and invite our first speaker, Dr. Sekharappa, sir. Dr. Deptt Patel, sir, the platform is yours now.
Thank you, Dr. Tushar Patel. It is my pleasure to invite Dr. Sekharappa. First of all, I am highly thankful to Dr. Sekharappa, sir, for your kind consent to accept our invitation for delivering the lecture on this virtual platform of the national webinar, Dr. Sekharappa, sir. Dr. Sekharappa is a professor of entomology in Institute of Organic Farming, US Dharwad, and graduated from the US Bangalore and post-graduated from US Dharwad. He started his journey as an instructor as agriculture entomologist in US Dharwad, and he is recently working as professor of entomology in the Institute of Organic Farming. He also worked as a head for All India Coordinated Sorghum Improvement Project for the period from 2012 to 2015 at US Dharwad. He visited various countries like Netherlands, London, UK, and he has guided 16 MSc Agri students and 2 PhD students. He has developed 12 technologies for the benefits of the farmers as well as scientific community. He is awarded by so many, he has also got the different awards, and among this, the most important, that is the champion of WSIS Prize 2018 from the Geneva, Geneva, Switzerland in 2018 for making the electronic solution for pest ESAP under the e-agriculture. He is also awarded by the Environment of the Year Award 2008 by National Environment Science Academy, New Delhi in 2008 for the work on biological control of insect pest. He is also involved in mass production of bioinsecticides, vermicompost, earthworms, and guiding organic farming in the cultivation as well as management of the insect pest of various crops. He is also associated with the subject expert for ESAP project that is worth 5 crore rupees. Also evaluated and identified the resistant sources and breeders materials against the key pest of sorghum and maize and developed the IPM technology for key pest. He has also published research papers in international and national journal around about 101, and also abstract presented in conference, seminar, symposium, that is 55. He has also wrote the 5 books and 7 chapters in the books, and paper presented in winter school, that is, and as well as summer school, that is 5, and he also published the popular articles as well as brochure, that is 63. So he has a very vast experience, particularly in the organic farming. So I'm highly, it's my immense pleasure to welcome Dr. Sekharappa, sir. Please, Dr. Sekharappa, sir, now platform is yours, sir.
Namaste. Good morning to everybody, and I'm very much thankful to the organizers for introducing me. Shall we start now, sir?
Yes, we can start. You can share your screen.
Okay, sir.
Okay.
ಬಂತು ನೋಡಿ ಸರ್ ನಿಮ್ದು ಹೈಲೈಟ್ ಹಾಕಿ ಹೇಳ್ರಿ ಫಸ್ಟ್ ತೋರಿಸ್ರಿ ಸ್ಲೈಡ್ ಬಂತು
ಮ್ಯಾಕ್ಸಿಮಮ್ ಯೂಸ್ ಮಾಡ್ಬೇಕಲ್ಲ
ಅದು ಬೇಡ ನಿಮ್ ನಡಿತೈತಿ ನೀವು ಮಾಡ್ರಿ
ಕಾಣಸಲ್ರಿ
ಇದು ಕಾಣತೈತಲ್ಲ ನೀವು ಮೂವ್ ಮಾಡಕ್ ಬರ್ತೈತಿ
ಅಲ್ರಿ ಮ್ಯಾಕ್ಸಿಮಮ್ ಯೂಸ್ ಮಾಡತ್ತಾರೆ
ಹಂಗ
ನೀವು ಸಣ್ಣ ಕಾಣ್ತಾರೆ ಹಂಗ
ಈಗ ಮ್ಯಾಕ್ಸಿಮೈಸ್ ಇಲ್ಲ ಬರಂಗಿಲ್ಲ ಅನ್ಸುತ್ತೆ ಸರ್ ಅದು ಪ್ರಾಬ್ಲಮ್ ಮಾಡ್ಕೋಬೇಡ್ರಿ
Yes, sir.
Yeah. Shall we?
Visible.
Yeah. Shall we start, sir? Now.
Yes. Of course, sir.
Okay. So, as the organizers has introduced about the today's topic, that is the management of insect pests, especially in the organic farming through myco-insecticides. So, the insecticides which have been originated from the fungal origin, so they are normally being multiplied under our laboratory condition, and they are being developed and they are being used both in laboratory as well as under field condition. So coming to the pest management, as all of we know that this is the basic thing. During the ancient times, we used to follow just traditional approaches for the management. We never used to have the especially the inputs like the fertilizers or chemical fertilizers or chemical pesticides or herbicides or whatever it may be. Then after the invention of the DDT, as all of we know that during 1939, especially that has entered from the medical field to the agricultural field for managing number of insect pests. So when this green revolution was started after the release of number of hybrids and varieties in number of crops, later on they were highly, as we know that they are highly responsive for especially the agricultural inputs, especially the chemical fertilizers. As a result of that one, the farmers used to harvest a bumper crop in the initial stage, and the insects which were there, they never turned as the pest. But in a few course of time, later on, these insects, they turned as a pest. They came in the way of the welfare of especially the farmers in obtaining a very maximum yield or very good yield. So after that, after post-green revolution period era, so many things happened. There was a catastrophe. Normally, the insecticides which were being used, especially in the management of insect pest, it was not in a sustainable manner, and they could able to, especially the insects, they could able to develop resistance. As a result of that one, the control strategies the collapse. It is mainly because of the farmers, they solely depend upon the chemical pesticides for the management of insect pest. So now we are in the gene revolution era, 2002 till this date. In spite of this also, we are not able to manage the insect pest through the chemicals or through these things. Just I will share those things in my future slides. As we know that for the earlier period was subsistence phase. Whatever the farmer used to grow, they were used to be happy. Whatever they used to get it back. And the exploitation phase, especially in the command areas where the irrigation facilities was there, all of these the agricultural commodities, especially the fertilizers, chemical fertilizers, and also the chemical pesticides, they were exploited in a maximum manner. And after a certain period of time, it was not sustainable, and immediately the yield, there was a drastic reduction of the yield because of the failure of the control measures, especially with respect to the insects as well as the diseases. And the farmers, they landed in the disaster, and so many suicides and so many things they happened in our country and also elsewhere. So later on, the people started thinking about the integrated control, as the chemicals can be used as one of the important components, not only the sole measure. So as a result of that one, now we are in the era of the organic or the natural control, because the people are very much fond of the agricultural produce or the horticultural produce which have been grown from the organic or the natural things. So these are all the things that has happened. One should not blame the chemicals, because they are chemicals. Definitely, they are very good, but they should be used in a very limited fashion. As all of we know that the salt is very much essential for preparing our dishes, but to what extent we have to use it, it depends upon us. That is very important. We cannot use it too much, and also we cannot use it very low. In a similar fashion, the chemical should be also used in a very, the limited things. So that would not happen in our the ecosystem or in under our Indian agriculture system, because there was a lot of gap between the extension functionaries and the farmers. As a result of that one, the farmers at the verge of greed to getting maximum yield, maximum profit, they solely depended on only the chemicals. As a result of that one, now we are facing a lot of crisis. As a result of that one, because of lot of the herbicides and also the chemical pesticides which are being dumped, it is going to create, as all of you know, that there will be air pollution, there will be water the contamination, pollution, and also the soil pollution. As a result of that one, the whole ecosystem has been collapsed. As a result of that one, so many diseases, not only to human beings, even to the domestic animals, wildlife, so many things are happening. It is mainly because of the sole dependence of the chemicals. As we all know that the ban of the endosulfan, and now the government of India is also banning number of chemicals because of their the ill effects, especially on the human beings and also the domestic animals. So whole our the microflora, fauna, and also the aquatic, the aerial and subterranean, all these things, the special beneficial fauna is going to reducing day by day because of the chemical pesticides which are being used, as we all know that. So this is a picture which just I would like to share is I am an endosulfan boy. So it is the problem which has occurred because of the aerial application of this endosulfan, especially for managing some of the insect pests. As a result of that one, it has reached the river bodies and also especially the water bodies. As a result of that one, the offsprings or the kids, they are getting such things. As the pesticides, as all of we know that they are going to contaminate not only the fruits, vegetables, even almost all the agricultural commodities, and we also have the ill effects, especially the applicators, especially the persons who are employed in application of the chemicals. They are getting more, because of their carcinogenic effect, they are also being severely affected. So these are some of the photos which are showing the ill effects of the chemicals which are being used. That is very important. So these are another pictures. A lot of our domestic animals, pet animals, even wildlife, everything there, and also the useful insects there, and also the microflora, they are being also, the vanishing day by day. So in spite of using so many, as we know that every year there will be a number of chemical, the insecticides which are being released in the market. So as we know that in spite of that one also, we are not able to manage the insect very satisfactorily. It is the main reason just now I have already told. Just you take the world, just pre-green revolution period, where these, especially the agricultural inputs, they were not being exploited too much. At that time, the world loss, the crop loss due to the insects, it was about 10.8. And after post-green revolution, it increased to 13.6. It means there was about almost 3% increase in spite of our several technology up to even the BT technology. So coming to the India, if you take this one, it has increased three-fourths. Just you see here, there was more than even 900 billion rupees loss is there as per the due to the insect pest. So it means what I want to say here is I'm not blaming the chemicals, but certainly they are good. But the problem is the time of application, quantum of application, how much to use, where to use, when to use, all these things. They have to be made very clear to the farming community. Then only they can be used as one of the components in the integrated pest management. Otherwise, it becomes very difficult, especially solely depending on only the chemical pesticide. That is my contention. So especially the underdeveloped countries like India, it is very difficult to educate the farms. That is the problem. So so it means we should rely very less on the chemicals, and we should go for other options of pest management tactics, as we have all of you know that. So this picture is, of course, it's a very common, and the losses caused due to the insect pest and other things are there. So what are the several other the strategies for management of insects? All of you know that there are physical methods of pest management, mechanical methods. Then also through crop habitat manipulation, also we can manage the insects very effectively. So as we know that the crop diversity, it is going to minimize the insect damage. So it is going to enhance, especially the natural enemies like parasitoids and predators, and also it is going to enhance even some of the entomopathogens, so that they can suppress the harmful insects which are going to occur on the main crop. So there are several options like intercrop, border crop, trap crop, ecofist crop, crop rotation, and also through plant nutrition and also through water management, we can manage the insect. And also giving the crop holiday, also we can break the carryover of the insect pest to the next season, and so that we can manage the insect. And also through the use of some of the resistant varieties wherever they are there, especially in the endemic areas, that can also combat the insect, especially in a particular locality. So coming to the other very important aspect is the biopesticides. As we all of you know that biopesticides, it may be of a plant origin or it may be animal origin like parasitoids and predators, and also the microorganisms like entomopathogens. So especially as we know that these biopesticides, which includes especially the fungal origin, microinsecticides, then viral, then protozoans, then bacterial, and so many things are there apart from the plant products and other things. But in spite of speaking all these things, biopesticides usage in India, it is still under infancy stage. Not at where it is less than 1% to say that one. So as per the recent data, the area under organic cultivation with these biopesticides, it is just 1.5 million hectare. It was during 2016, and now it has been increased to 1.9 million hectare. But because of the people awareness and slowly it is increasing, but it cannot be compared with the other, that is the chemical method of the approach. So out of during 2016, the biopesticide consumption was just recorded as 15%, and rest of the 85% it was only again the pesticides or the chemical aspects. So just even now also we are depending heavily on the chemicals. That is the problem. And the consciousness of organic farming and use of biological origin product is on increase. Now the farmers are slowly they are moving towards the non-chemical approaches. So a lot of the commodities, especially the plantations commodities, or it may be the fruits or vegetables, or even the agricultural commodities, which needs to be exported, and definitely certainly they need the organic approach for the management. So there is a slow increase in, especially even in the Europe and other Western countries also, as per the WTO agreement, as per the free trade, normally, the pesticide-free commodities are very much essential. That is very important. In the last 10 years, especially the consumption of biopesticides has increased by 23%, while that of the chemical pesticides has grown only to 2%. That is very important, and it is very, the thought-provoking, and we should concentrate. That is very important, and we should encourage the biopesticide. That is very important. And not only in Europe or Western countries, and especially USA, and also in India, number of chemicals are being also banned because of their ill nature and other things. So especially the green label molecules, especially can be effectively used for managing insect. That is very important. So of course, coming to the biopesticides, as we know that they are microscopic, microscopic, and phytochemicals are being, just now I have explained. And as for the today's the talk, as per I am going to talk only about the insect pest management through the fungal origin. So the another speaker is going to speak on the entomopathogens management in the subsequent the lecture. Okay, so these are, of course, as you know that as do we get the diseases, the insect also gets the diseases through the viruses, bacteria, fungal, and also the protozoans and nematodes. So they can be, naturally they are occurring. Nobody has the developed them. Just they are occurring in the nature. We are going to harvest, or we are isolating them in the laboratory condition and multiplying them and making use in the agricultural ecosystem or the horticultural ecosystem, because especially because of the usage of these broad the pesticides, normally these the beneficial organisms have been diminished like anything. That we would like to regain in the nature. That is very important. So coming to the why these fungal, there are about different classes of fungi, but of course the most important things are the fungi which belong to the Detromyces, especially the fungi imperfectae. They are very important because they are facultative and they can be multiplied very easily under laboratory condition, not like virus. They need a specific host for multiplication. In case of this fungi, they can be multiplied artificially on in laboratory, under laboratory condition. That is the beauty of that one, and they can be utilized under field condition for management. That is the very important. So under in the Institute of Organic Farming, we are multiplying in a very large scale. That is especially the Metarhizium, then Nomeria. That is Metarhizium delay. Now it is being called. Then Licanicilium licanae. These four, the Beveria also, they are being multiplied, and we are giving to the farmers just for on a subsidized basis for encouraging the organic growers with respect to the pest management. So coming to as I said, what is this Metarhizium supply? They are very effective again, especially the soil inhabiting the pests, especially like the termites, then the root grubs, and also some of the sucking insects like the roam plant hopper or white back plant hopper, and so many things are there. Like Beveria bassiana, it is also very effective against number of the homopterans, lepidopterans, coleopterans, and also so many things. And especially this Licanicilium licanae or the Verticillium licanae. So they are very effective against sucking insects, especially the soft-bodied insects like the aphids, thrips, mites, and mealybugs, and so many things. So coming to this Nomerillae, now the name has been changed to Metarhizium rillae. So they are very effective against lepidopterans insects, which are mainly the acts as a defoliator, and also the pod feeding or pod borer or whatever it may be. And another important aspect is Hirsutella thamsane is very effective against mites, as we know that the perianth mite, especially on the coconut, is being managed with Hirsutella thamsane. But it's mass multiplication we are not undertaking, but we are taking only above 4 things. So these micro-insecticides, why they are very important, that is very important. Yeah, that we have to look into that one, because as we know that they are very specific and they are not going to cause any the side effects or especially to the non-target organisms like especially the beneficial fauna. And the development of their insect resistance, it will be very slow or will not be there. That is important. And another very important aspect of this the fungal origin insecticide, especially the micro-insecticide, is the epizootics. Only thing is these since they are also living organisms, the congenial climatic conditions like the high humidity and the optimal temperature like 24 or 25 degrees centigrade is very important. Just by crop habitat manipulation or through the irrigation, and in certain pockets we will get certainly these type of the climatic conditions. There we can utilize them or exploit them for managing the crop pest. Especially whenever we are going to apply, they are going to invade these insects and cause the disease. As a result of that one, they are going to be killed, and millions of conidial spores they are going to be developed. As a result of that one, upon drying, those cadavers they are going to fall on the soil through the wind and also through the irrigation water or the rainfall or so many things. They are going to be disseminated in the environment. Otherwise, they can also persist in the soil. In the next season, their persistence, it may be 50%, 40%. So that 30 or 40% it will take whenever these congenial climatic conditions they are going to occur, and whenever, even if you are not going to apply the next season, so whenever the pests they are going to occur, normally they take care to manage those insects. So what we call it as epizootics. So it means once we apply in the environment, if they are going to persist under the favorable climatic condition, definitely they take care of the insect pest in near future, whatever the agricultural crops or the horticulture crops we are going to take. It means there will be a sustainability in the management of insects, so that we are going to introduce or pump the beneficial fungi in the environment, so that we can get a very remarkable control for the insect. And they are compatible with other tactics of pest management. So normally, except the chemicals, we can integrate these things with other under IPM program, and we can also manage. For your information, to develop one, the micro-insecticide or biopesticide, normally it may require just around 5 crores. But to develop chemical, this thing component, it requires 500 crores. But in case of, as we know that the recurring expenditure, in case of especially these biopesticides will be very less, whereas in case of chemicals it will be, as we know that the chemicals which are coming today, it will not appear after 2 years. So it means they are not sustainable, and they're because of the development of resistance and also problem, normally they vanish from and every year there will be development of these things, and also the people are not just they forget about that one, and normally another thing. And another thing, as you know that they are highly potential for further development through the biotechnological approach. So that especially these fungal origin, the micro-insecticides, what we are telling, we can increase their the virulence, especially these different isolates through this biotechnological approach, so that the mortality rate or the under field as well as under laboratory condition can be enhanced through this biotechnological approach, and there will not be any threat to the biodiversity because they are very or highly host specific. That is very important. So these are the slide shows about the commercially exploited these very important the micro-insecticides, and they are being marketed under different trade names under Indian situation. But if you take the global, this thing, there are plenty of the branded, the micro-insecticides are available. So normally they are going to cause the mycosis, and of course out of 950, 10 species have been exploited, but 20 have been right now have been exploited in the integrated pest management. But their availability is very important, because normally the mass production of these things is not taken by the private entrepreneurs. That is there may be a little, but they are very in a very very limited fashion it is there. But especially, in the public, in the public domain, it's very important to enhance the mass production of or availability of these things to the farmers is very, very important. So especially this metarhizium supply, it has been detected to cause disease, especially in 300 instances. Just to imagine the very, very important. So they can be exploited very well, especially for the root grubs and beetles, termites. And in the future slides, I will show you, especially the soil dwelling insects. Especially, it becomes very difficult to manage with the chemicals, as you know that one. So because unless the insect comes in contact with the chemicals, they will not be killed. Whereas in case of these microinsecticides, what happens if we apply through some organic manures? Normally, the grubs which are inhabiting in the soils through the irrigation water or through the cultural operations, and also the insect migration, they comes in contact with that one. Once they come in contact with that one, as I said, millions of conidia they are going to spores they are going to produce, and as a result of that one, the whole the soil is going to be mixed up with these cadavers, the conidial spores. So as a result of that one, they are going to take care of further this thing. So whatever the quantum of these insecticides we are going to apply in the soil, normally it will be multiplied n number of times. That is very important. So they are also being used in more than 15 countries. Of course, lichen silica, as I said, soft-body insects, metarhizium, same thing. And the Bevirea bassiana can be also used against some of the very important these insects under plantation as well as the horticulture crops. So as all of you know that, of course, this is a very basic thing. So these are especially the fungi and also the nematodes. Normally, whenever they come in entomopathogenic, these nematodes and also the fungi, whenever they come in contact with the insect, just they gain in the entry inside the host body and they multiply and they just, they bring the death of the host insect. Whereas we can call them as a penetrating microbes. Whereas in case of virus and other things, normally it is very difficult because, they have to be ingested through the food. But here it is not the case. Just by contact, they are going to cause the disease, and we are going to get a very satisfactory control. So as a result of that one, as all of you know that the insect which has been infected with these microinsecticides, they lose the appetite and there will be irritability, and also the paralysis. We can also generalize the paralysis we can see, and discolor the integument patches, white patches we can see, and the insect cuticle becomes hardens, and the insect, it is going to be mummified, and it is going to, with the upright position, just, it is going to adhere to the substratum or the plant parts which are going to be there. That is very important. And the insect death occurs with normally in these within 6 to 7 days, or sometimes if it is highly congenial, based on the virulence and the environmental condition, even the death can be also noticed within 4 days also. So these are the different insecticides. I mean, the microinsecticides, they are being used in different the insect pests, like the white grub, banana rhizome weevil, sweet potato weevil, brown plant hopper, as I said, then diamondback moth, rhinoceros beetle. These things can be managed through application to the soil by mixing with vermicompost or with farming manure. And similarly, the Bevirea bassiana, Licanicilium licanae, metarhizium, they can be applied either in the spray formulation, and they can be effectively used for managing number of insects. So this one thing, and this picture shows about the numerial, that is the metarhizium. Now the range name has been changed. Just to see here, in case of the chemicals, as you know that we cannot virtually see the death of the insect. We can see the reduced level of damage or infestation, but you cannot virtually, it becomes very difficult. Whereas in case of entomopathogens like even the virus and other things, bacteria, you can see virtually the death of the insect. Even in case of the microinsecticides also, once the insect comes in contact, you will see the cadavers, dead cadavers adhering to the different parts of the plant wherever they are attacking. Just you see here, in case of cotton, this is the Helicoverpa parmigera, this is the Spotaphyllitra in case of groundnut, and this is the semilooper in case of green gram. And likewise, in case of chili, Helicoverpa parmigera, then again the Helicoverpa parmigera in tomato, and spinged caterpillar in case of the sesame. And upon after attaining the death, after a certain time, because of the jerk of the wind and other things, the cadaver falls to the ground, and you can see the lot of these conidial spores. They are coming in contact with the soil. As a result of that one, in the whole the ecosystem is going to get epizootics because of the death of this insect. It means just a single spore is conidia is sufficient to cause the death. But you will find a lot of the millions of the conidial spore. I mean the spores which are going to be pumped in the environment. That is very important. So it can be used in very, very effective, and some of the experiments conducted very long back. Of course, we have already made recommendations in number of crops, especially in our the US the Harvard jurisdiction, where the congenial climatic conditions are there, like especially it comes under transitional tract. We have the high humidity and the optimal temperature. So they can be either mixed alone. I mean, they used alone, or they can be used in integrated fashion, like the neem seed kernel extract. Then some of the plant products can be used in the as a biodynamics, and we can apply with the Navsari relay at the rate of 2 into 10 to the power of 11, or now we are recommending at the rate of 2 into 10 to the power of 8 the CFU per gram substrata. So this can be very well used, especially it can be also followed with Panchagavya. So these are all the organic treatments. This is, I mean, they are going to yield much better than the chemicals alone using. That is the important, and the damage, just you see the pod yield and other things, especially in case of groundnut, we have achieved a maximum yield. So without damaging the ecosystem, we can use these things. Only thing is we have to be very patient in doing all these things, and we have to this thing. And in case of potato, so same things they are also there. The recommended package, of course, it has yielded 4.21, but the NSK is top. And coming to the next best, they are seem to be the again our micro-insecticides for managing the insect. So these are the same picture. Some. And as I said, they can be used, and just we are making just a temporary the pouch for just to supply to the farmers for to use or to encourage these the micro insecticides under the field condition. So similarly, we can also use especially the see these are the closer pictures of these insects which have been dead due to the Navsari relay. Same thing. So the same thing, same pictures in case of sunflower, the other crop extensions like the okra, then green gram, tomato, sorghum, then pigeon pea, then the chili, and also the marigold. Like that, in number of crops, so we can use even in chickpea also, so that they can take care of the insect. So the same, the Spodoptera litera has been infected with the Navsari relay upon spray. Just you see the fallen cadaver on the ground just you after the debris, just you see that. So they will be remaining in the environment in the subsequent, the season. Definitely they are going to take, especially for controlling or managing the insects in the subsequent crops. So these are the things even in case of the potato, and also the fall armyworm in case of maize, which is occurring a new pest. And we are also supplying to some farmers for see we are not able to meet out the demand, just they are taking care of this one. As we know that especially for fall armyworm management, normally we are recommending some of the ad hoc recommendations like some chemicals, the spinosaurum, chlorantraniliprole, and other things. But the farmers, even after the repeated application of about 3 to 4 applications, they are unable to contain this pest. So the Navsari, they are highly beneficial, and the farmers which have taken long back, they are also using those things, and they are and telling that this time we have not used any spray of this Navsari, but we are able to get the infection. So this is how they are going to cause the disease. That is very important. Even in case of course, especially in case of casters, especially for the Spodoptera litera. So Vimala Devi and Prasad very long back in the Andhra Pradesh from Hyderabad, they have reported the efficacy of this one on the castor, especially the Spodoptera litera. Even in case of soybean, Spodoptera litera, it has been also worked by a number of people. In our department, we had a very the NATP project. Under that, our ex-director of research and entomologist, senior entomologist Dr. Lingappa and his co-workers, they have worked on this one, and they have also brought out a number of brochures, and also they recommended this particular micro-insecticide for managing number of crop pests, especially managing defoliators and also the pod borers. So this is one thing. And the next is so this is the container that how we are going to make use of that one, and just we are at the work of now the registration of this one. Now we are working for that one. We have obtained the accession number, and we have obtained the toxicology data from the Patapai, and so many the requirements for registration of this particular myco-insecticides. We are at it, and just we are taking the help for some organization for taking the bioefficacy data across the nation, and soon it will be submitted for registration, especially this particular fungus, that is the Metarhizium relay. Now it is being named. Okay, so if you take the Metarhizium menisoplex, just you see here the extent of the pathogenicity can we see here. Just you see here, it has been tried under laboratory condition from this department only in our institute. And American bollworm, this Metarhizium menisoplex is very effective. The coconut black-headed caterpillar, just you see the mortality rate under laboratory condition. So it means they are certainly they are going to cause they are highly pathogenic to very noxious insects, and they can be used very well wherever these insects they are going to occur. That is very important. So they are also effective against not only to the lepidopteran insects, they are also very effective against the coleopteran insects. So that is very important. Just some of the photos, just you see a Helicoverpa armigera infected with Metarhizium menisoplex, and the cowpea pod borer Maruca. So it has been infected with Metarhizium, just you see here. So that is very important. And in case of sugarcane, especially the root grubs, they are going to be effectively managed. We are especially under our sugarcane belt. Normally, the farmers they heavily depend on Metarhizium menisoplex. We are recommending at the rate of 5 kg plus 500 kg of FIM or the vermicompost that has to be applied to the soil during June, July, and also during December, January. So that during this both type of plantings, in case of sugarcane, they are able to cut. Otherwise, the farmers what they used to do, earlier, the chlorpyrifos, forate, then carbofuran, which are the verge of fanning, all those things they used to dump. They used to apply 10 times, 12 times, just you see the ecosystem, how much damaging, especially in case of sugarcane, the biological control, especially managing the insects through the non-chemical approach is one of the successful example. And in this case, we are in our particular geographical area, where
Dr. Sekhrappa sir, please unmute yourself. Dr. Sekhrappa sir, please unmute yourself. Your voice is not here over here. Dr. Sekhrappa sir, please unmute yourself.
okay.
Yeah.
Now
you are audible.
Okay. Okay, so as I was telling about the Metarhizium supply, it is highly effective to number of insects, both lepidopteran, coleoptera, and so many things. And also I was telling about especially the management of this one in case of especially brown plant hopper, in case of paddy, especially this brown plant hopper. As you know that in case of transplanted paddy, normally you will find high humidity as well as because of the. Okay.
Please full screen your.
Yeah. One minute. Just a minute. So now it's okay.
Yes. Now it is.
So in case of paddy ecosystem in the command area, especially in the transplanted paddy, normally we have the high humidity as well as the congenial climatic condition, especially the temperature which will not cross 24 or 25 degrees centigrade. This brown plant hopper, it is very difficult to manage because in our ecosystem it has become endemic in certain areas, both BPH as well as the WBPH. So to manage this one, this Metarhizium menisoplex being used at the rate of 2 into 10 to the power of 8 CFU per gram. So plus 0.1% twin 80 that is being used. So whenever we are going to apply it to the base of the plant, just above the water level, especially these the hoppers, they get infected with Metarhizium menisoplex, and they are going to cause the mycosis, and we can see the very good effective control that one. And coming to the red gram shoot weevil, especially, it is very difficult to manage with any sort of chemicals in the. They are going to form the gall in the red gram, and the grubs which are seen here, it will not. It is very difficult to come in contact with the chemical. So we apply this one, it definitely is going to take care of that one. That's why this is very important to manage this certain noxious insects. And some of the field studies which have been conducted in chickpea and other things, just you see here, Metarhizium menisoplex, which has been used in the spray formulation, it has effectively controlled especially the Helicoverpa armigera, and the incremental benefit cost ratio is about 8. It is it is very high and uncomparable with especially the recommended package with the chemical. So that is the thing. And similarly, we can also manage especially especially we can also manage the white grub, especially in case of sugarcane. We have used it especially in sugarcane belt area, and this Metarhizium menisoplex is most effective as compared to the chemicals. Very important. So that can be exploited. That's what I was telling you. And similarly, we can also use this one, especially in case of this is in the other location where we have recently conducted some experiments, not only during 2013, even last year also we had a very large scale the demonstration these things. So I used different strains of this one. Here also they have proved very effective in giving maximum returns. But maybe after the chemicals like chlorpyrifos and anaxifer, but in a sustainable manner, in a long range, we can manage these insects. That is very important with this the eco-friendly or green labeled insecticide. Especially in case of paddy, I was telling the combination of T1, T3, and T1, it is found to be very effective, so that the Metarhizium menisoplex can be integrated with other these things and can be utilized as a sequential spray for managing that one. And the Acidotus coleus, just I have shown you in the picture. The similar case is here, but certainly they are not as effective as the chemicals. But in due course of time, if you use in a very long range, definitely they are going to take care of the insects. That is very important. In case of the copy is also, the results are not that encouraging, but certainly there is a scope for using this Metarhizium menisoplex. And some of the rhinoceros beetle and Orycters rhinoceros, they can be very effectively managed when we use at 5 to 10 to the power of 11, and it has been reported very long back, 2000. And from our own the studies from our institute, the white grapes can be very effectively managed, and but it is found very next best to chlorpyrifos. So it means there is a lot of scope for managing these things. Even Nahar from 2004, they have reported the mortality rate, especially with Metarhizium menisoplex. It is very high in the Metarhizium menisoplex as compared to the endosulfan in Maharashtra. And similarly, the Bhagat from white grub in case of potato, they have also reported encouraging results as compared to the chlorpyrifos. So these are some of the very encouraging results. Even the Pandey from especially for soybean in case of white grub, this can be very effectively managed with the Metarhizium menisoplex. So this photo shows about this is the groundnut leaf miner. This can be very effectively managed with the Metarhizium menisoplex in the form of spray formulation. In case of banana, the rhizome weevil can be very effectively managed by mixing with the vermicompost or the fermented manure. And even the termites can be also very effectively managed. These are the root grubs, the adults, of course, as I've already shown in the sugarcane ecosystem. Especially in case of the mango leaf hoppers, especially the organic mangoes, where the number of farmers they approach for export quality. In that case, for managing these leaf hoppers, normally we recommend Metarhizium menisoplex spray, especially at the time of 25% flowering, 2 or 3 times, so that we can effectively manage the leaf hoppers in case of mango. Of course, this is there, that is the root grub in case of sugarcane. Okay, so these are some of the insect pests which are highly susceptible to the metarhizium supply. Just to have already talked, this is the coconut black-headed caterpillar, because it's very difficult. But you know, just if you apply this insect, I mean the micro insecticides, they will take care of the pests which are going to occur in the film. Even in the forest ecosystem, they can be introduced very well, because it is very important that especially the teak defoliator. So in order to manage that one, we can take care of with the metarhizium supply. Of course, these things we have already discussed sufficiently. So let us move to that one. Same thing, this paddy brown plant tapper, that is the infected cadaver, just you see here, and they can we are selling to the bird. And coming to the sheep infected with the blue tongue virus, that is very important. So this is going to be transmitted by some of the flies like Pelicoides, nubeculosis. So especially they are found in the dung of that one, and they are going to transmit the blue tongue virus in the cattle. So this particularly this fly can be managed effectively, especially in Western countries with the metarhizium NA supply. Just you see here. Just you know, see here, it has caused about 100% mortality when they are going to use a strain like wheat 279. It has been also commercially exploited, and the transmission of that blue tongue virus has been effectively managed by especially in USA. So especially this is this fungi will not pose any risk, especially to the wildlife, including human beings, birds, fish, and other things. Just they can be used especially even in case of poultry manure. There will be a lot of problem with this the house flies also there. So in some of the village nearby our you know the area, where these poultry farms are there, the farmer, I mean the villagers, they used to put in a heap, especially this poultry manure. So there was a lot of these poultry manure. Normally they act as a breeding site for multiplication of especially these flies, especially house flies. So they can be very effectively managed by application of this, you know, the metarhizium supply, because this is normally metarhizium supply, you know, they multiply at a very faster rate in, especially in the organic manures. When there are the grubs or the maggots of these flies are there, they are going to be infected, because normally the root grubs, they feed on the organic matter. When they feed, you know, normally they get be killed. That is very important. They are being also used, and the efficacy of this, you know, the metarhizium supply can be also increased, especially the scorpion species. The especially this the A. australis, this particular species is used to increase metarhizium supply's insecticide strength by 22-fold. So this toxin is used especially from, that is, University of Maryland, and after using that one, you know, that becomes the hypervirulent fungi, and that can be used as a insecticides, what they are reporting. Especially, you know, from the scorpion, you know, the neurotoxin is extracted, and with that one, the DNA, recombinant DNA is formed, and normally this the DNA is inserted in the fungi species, and it is especially bioengineered with the help of the clone of this metarhizium supply, and they are naming that one as the AS549. And especially when they are going to this AIIT is activated, and whenever it is going to be transferred to the bloodstream of the insect, so it means they are going to bring the mortality of that one. So this is how they are being, you know, with the help of this recombinant DNA, they are also, you know, the using or increasing the virulent power to up to 22% or the 22-fold. So coming to the Licanicilium licanae, it is very well documented, long back only reported their efficacy by different workers across the nation, as well as from the abroad also. So especially for soft-bodied insects or the sucking insects like the coffee green scale, then the citrus green scale, and Indian mustard, rapeseed, they can be very effectively managed by spraying these the Licanicilium licanae under field condition. So in South Korea also, it has been also used especially for so the white flies and also the green peach aphids for managing the insect. And especially even in our university also a lot of work has been done with managing especially the sucking insects with Licanicillium licanae number of crop ecosystem. Okay, so this has been also reported by Kerry et al, especially for managing the cyst and root knot nematode. That is also very important. Coming to the reports of natural infection of this Licanicillium by different workers. Just you see here, the lot of aphid species are there, then scales are there, then so many the sucking insects can be very effectively managed, and their pathogenicity and effectiveness has been well proved by a number of workers across the nation. Just you see there is a big list just I have compiled, and there is a lot of scope, and even in our area also, especially the Licanicillium licanae is being multiplied and given to encourage especially the managing, especially the sucking insects in case of number of crops, even including mulberry also, because in case of mulberry, the thrips is a major problem in our area, where we are advocating the farmers to use this Licanicillium licanae, because the mulberry leaves cannot be sprayed with the chemicals. Otherwise, they have to wait for a very long period of time for not going for feeding, especially the mulberry leaves to the silkworms. So that's why if we spray this Licanicillium licanae, we can effectively manage that one. And even in case of the mango orchard, wherever this the mulberry intercropping plantation is also there, the organic growers are there, and they have to manage leafhoppers in case of the mango, and also in case of the mulberry. In that case, we are not recommending metarhizium insufflare for managing the leafhoppers in case of mango. We are recommending the Licanicillium licanae, so that they can effectively manage both the leafhopper in case of mango and the thrips in case of mulberry. So likewise, there should be some the alternative strategies we have to think, and we have to manage very effectively. It means what I am want to say here is, there is a lot of scope that these can be exploited on a number of host insects under a different ecosystem, unless the ecosystem has to permit. That is very important. That is the congenial climatic condition. So this is how it is being also reported by several other workers. Just you see here, number of insects, just I have compiled very important and very few of them. So these are the photographs which are showing especially the aphids. They can be very effectively managed with this, the coccids and other things, scales. So very effectively with this Licanicillium licanae, very effectively. So this is also being used, and there are also some commercially available, some of the Licanicillium licanae products are also there. They can be also used. Coming to the Beveria bassiana, it is going to cause disease, especially in 200 species of insects which are belonging to 9 different orders. That is, it has got a very wide host range. And normally the leaf hoppers, plant hoppers, whiteflies, caterpillars, diamondback moth, coffee berry borer, especially the coffee where it is being exported, coffee berry borer is a very severe problem in our ecosystem. The coffee berry borer can be very effective. Some time back, the coffee board has approached us, and we had supplied this Beveria bassiana. And this Beveria bassiana is also very effective in most of the gramineous borers, especially the cereal borers, like the stem borers and other things. So this can be used very effectively for managing, even in case of sugarcane borers and other things. That's very important. And even for the root grub also, it is very effective. And there are several reports which are saying the mortality both under laboratory condition and also in field condition. So even for managing rice hispa, that can be also is a recent problem, especially in some of the paddy ecosystem. It comes in a very epidemic form, and there we are recommending this Beveria bassiana. Similarly, even in long back only, it has been reported from Coorg that can be also used, especially for the coffee berry borer, as I said. So these are some of the workers which have used again as some of the insects for managing, especially this Beveria bassiana. In case of sunflower, and in case of the tea, that is the tea looper and green ram, white grubs, and so many things. And especially in the abroad, the Beveria bassiana is being used enormously for managing the root grub. It is almost on par with the metarhizium insufflare. But only the thing is, the isolate or the variations or the efficacy of the virulence or the strains is very important. In a particular locality, some strains or the isolates, they will be very effective as compared to the other geographical area. And the one which are going to perform well under the Harvard condition may not be suitable in Pantnagar or in Chennai or somewhere else. They may be suitable or may not be suitable. Only thing is the local isolates have to be taken into consideration, and they have to be isolated, and they have to be multiplied, and they have to be used for against the by various kinds of the insects. That is very important. It is just like in case of cricket, Ranji, in their home pitch they will perform better, and if they go outside, normally they don't perform well. In a similar fashion, it also holds good here also. That's why, because of it is mainly because those isolates, they will be coexisting in that particular ecosystem very long period of time. So it means they know the weak points of the host insect and also this thing. And that particular ecosystem, generation after generation, years after years, they are coexisted, and as a result of that one, they can bring down they can suppress the population or the menace of the insects. That is very important. This is the coffee berry borer, what we have seeing here. And there are also some of the commercial formulations of Bavaria bassiana, like Larosil. Sometime back, this the Excel company they used to produce and sell, and our own university is also producing. And red palm this is a very severe problem. That can be also used very effective, can be managed with this one, and diamondback moth, and also the other the coleopterans. So this is how they are being used. And as I was telling, the poultry farms, the metarhizium supply, and also we can use the Bavaria bassiana in the management of especially the flies, which are a big menace in case of cattle. So especially the poultry manure, we will find a lot of these the flies, which as I was telling, the villagers, they used to dump this poultry manure in large quantity. They even it was multiplied in a very exponential manner, in such a way that even they were unable to drink the water just because of the flies they used to get inside the water. It means so in order to manage that one, under such situation, this the chemicals will not come into help because of the continuous application. So in that condition, metarhizium supply or the Bavaria bassiana can be mixed directly into the poultry manure, and we can bring the death of these maggots as well as the flies which are going to occur. That is very important. So under that situation, it's very effective, especially it is being used normally in the Western countries and other things. So of course the coconut mite, of course the perianth mite, the mycohit, it was the marketed by some time back IHR, and the Histotella thomsoni is also found to be very effective, and this can be also exploited to the possible extent, because even now also this perianth mite is a major problem. It is still a burning problem, as we know that especially we have to take care of management aspects. Under that situation, the perianth mite, as you know, that this mite will be beneath the perianth, and whatever the insecticides we are going to apply, it will not come in contact. So because these tender coconuts, they are being consumed, as we know that one, and in order to manage this one, this the Histotella thomsoni can be, but we are not working on that one. And this Bavaria bassiana, these are also very effective against black or brown fire ants, cockroaches, termites, and so on and so forth. Okay, so this is very important, and especially some problems there. Okay.
Can we conclude our procedure?
Just 1 minute, just it has delinked. Okay.
Is there any problem, Dr. Sekrappa sir?
Yeah, just little, just it has taken to the internet.
If any problem, then we can share the screen from here.
Yeah. Share it, sir. Yeah, 1 minute, just no problem. Okay, so these are some of the insects. Can you hear? Audible?
Yeah, you're audible.
So they can be used very effectively, and these are just as reported by Ramanujan. long back, that is 2014. Just Bevere Bessina is found to be very effective under different brands of this one, and these are the different the target pests.
Screen is not visible. Dr. Sekharappa sir, your screen is not visible.
Just 1 minute, sir. You can share, sir. Just
yes, we share your presentation over here. Don't worry.
Okay. So in another 1 or 2 slides are there. Okay.
Okay. Don't worry.
You please share, sir. Okay. Yeah.
Okay, visible. Is it visible?
Yeah. It is visible.
Okay, go ahead, sir. Please.
Okay. So as we have talked much about, I think a lot of information is there. Just I have tried to compile only a few information. You know, the truckload of information is available. The only thing is we have to exploit them in a very better manner for according to our geographical area. That's very important. And in spite of speaking all advantages of these the biopesticides, still we have a lot of the disadvantages or the constraints. As all of we know that there is a lot of the lack of knowledge, that is very important, and it can be used in the integrated pest management, and only a successful candidates are on sale, especially because they are not available to the farmers. That is very important. And especially the distortion of the information during transmission, of course it is everywhere, and non-adoption over the larger areas, that is very important. Then inconsistent and the unplanned demand for the bioagent, and they have got the very short shelf life, that is very important. As I said, they are climate dependent things, and they are highly dependent on the climate. Only the under climatic the congenial climatic condition they can work. Next, sir. Next. So they can work only under very limited this thing. Next slide, sir. Hello. Shall we move to the next slide, sir? Hello. Shall we move to the next slide, sir? Is it audible? Okay. So there are certain steps are required to boost the biopesticides use. That is the first thing is lowering the product cost. That is very important because it requires a lot of human resources as well as the inputs for manufacture. And the insurance of the quality product is very, very important. That is very important because they require for storage, very good, the whatever the conditions they are existing in the lab. That is very important. And of course, the creation of awareness is there. Then simplification of pressure. This is the very biggest hurdle in our system because for registration, it has been made on par with the chemical pesticides. So it is very difficult to get the registration for especially the procedure. There are a lot of problems or the hurdles are there. So that procedure has to be simplified, especially for the government organization. That is very important. Then the incentive to the farmer. That is, they have to be made on subsidized rate, and the farmers has to encourage, and they should stop discouraging. Or they should discourage, especially giving subsidy to the chemicals and other things. So that these are some of the important things we have to look into that one. Next, sir. Next slide. Next slide, please.
Okay.
Yeah. So this is what I wanted to tell about the use of these myco-insecticides in the management of insect pest. So this myco-insecticide, just we have seen very little. Likewise, we have the viruses, and the next speakers they may talk about all those things. And protozoans are there, bacteria are there, so many things are there apart from the natural enemies like parasitoids and predators. So it means there are a lot of the ample of opportunities we can look for managing the insect. No need to solely depend on the chemicals. That is what I wanted to tell. If at all we have to use them, we have to use them in a very integrated manner and in a very limited manner. That is very important. So with this, just I take this opportunity to thank the organizer for giving me an opportunity to share just a very little information with you all people. And now I request all of you to interact if any suggestions or any the clarifications required from my end, just I will share with you people. Thank you, sir.
Yes,
Yes, now the topic is open for discussions. Participant may interact with Dr. Sekharappa, sirs. Or you can also put the question in chat box. I think Dr. Sekharappa, sir, we have received a complimentary message for you, sir.
Okay,
participants have mentioned that the presentation is excellent. You have deliberated things very comprehensively and resourcefully, sir. So,
okay, thank you, sir.
So without further delay, I just again welcome those who are just joined the sessions. Especially, I observed the presence of Dr. Anil Dixit. He was one of the resource persons of yesterday's session, sir. So I welcome you, sir. Now I invite Dr. J.J. Patel, sir, to propose a vote of thanks to Dr. Sekharappa, sir.
Okay, thank you, Dr. Patel. Thank you, Dr. Sekharappa, sir.
Thank you, sir. Thank you very much, sir.
In organic farming through microinsecticides, it's a very important topic. Even your presentation is very nice and informative. Before the era of biopesticide, there is only key factor, that is use of insecticides. It is only effective for the insect pest management, but indiscriminate use have so many problems like 3R, that is resistance, resurgence, even residue. So many insecticides have been banned as they are poison for fishes, bees, pollinators. So it also targeted to beneficial insects. So the biopesticides like plant products, parasites and predators, and pathogens are very effective, and it is a new era for the pest management. Even area under organic cultivation is also increased with the use of biopesticides, because they have rich market when we use green label markets. So it is very rich. Even microinsecticides are prepared from virus, bacteria, fungi, protozoa. So they are well known examples for control of the insect pest. Second thing, it is very safer to non-target beneficiaries, and it is highly specialized and effective. Some constraints like lack of knowledge, suspicion, not adapted for the large area. Even we are not providing the subtle demand that's made from the farmers. So these are some constraints. Overall, it's a very good topic. So I congratulate Dr. Sekharappa and thank you so much.
Thank you, sir. Thank you very much.
Thank you, Dr. J.J. Patel, sir. Now I invite Dr. Swapnil Deshmukh ji for introduce our next speaker, Dr. Swapnil Deshmukh ji.
Thank you, Dr. T. Patel, sir. Now our next speaker is Dr. Lalit Mahatma, sir. I am very happy to introduce him to our participants. He is a very eminent personality of our university. He is Doctor of Plant Pathology and also Fellow of ISMPP. He is presently working as an Associate Professor in Navsari Agriculture University, Navsari. He has a vast experience of 17 years in this, with the main research of plant viruses and biofertilizers. It will be not false that if I mention that because of his efforts, our university has an upper hand in South Gujarat in case of biofertilizers. He has guided 7 PhD students and 14 MSc students. And presently he is also he is having 6 students for PhD program. Our university had a very recognition in case of this research, development, production and distribution of biofertilizer with the technical guidance of Dr. Lalit Mahatma sir. Now he has also awarded with the Best Teacher Award in 2014 from the Navsari Agriculture University. He is also having this Best Young Scientist Award, that is Professor H.C. Dubey Outstanding Young Scientist Award. Then BP Single Memorial Pesticide Industry Award, and Excellent Performance Awards in many of the symposiums and national seminars. So I welcome you, sir. Now, sir, the platform is yours. Dr. Lalit Mahatma, sir. Sir, you can unmute yourself.
Yes, I am audible, sir.
Yes, yes, you are audible, Dr. Lalit Mahatma ji.
Can you see my screen, sir?
Yes, yes, your screen is visible. Please do your presentation in full screen.
Yeah, could you see this blank slide, sir, here?
Yes, next slide. Yes, yes.
Thank you
Yes.
very much. Thank you very much, sir. Thank you very much, sir. Thank you, Swapnil, for introducing me. At the outset, I would like to express my sincere thanks to all the organizers under the leadership of Dr. K.G. Patel, sir, who is organizing this seminar and has invited me once again to interact with the scientific fraternity. Sir, I have seen splendid team building capacity in our respected Dr. K.G. Patel, sir. And because of this, like Bharuch, College of Agriculture, Bharuch would organize is many seminars during this COVID period, which is count in dozens, isn't it? So if I will use the word like one seminar, two seminar, 5 seminar, 10 seminar, then it would not justify the way in which you all people have done it. It counts in dozen, if I'm not wrong. I remember very well because I know him very well personally also. I'm also of the same university. So I know him very well personally. And here I would like to remember my early days of interaction with Dr. K.G. Patel, sir. Sir, I saw when I came to Bharuch and interacted with you during 2012 and all these things. You were the single person. You are the only person there. And you were trying like the best anybody can do to develop this center. I remember at the time I used one word at the time, that one-man army. Soon, because of your team building capability, it was happened like this when I visited. Next time, I think I remember it is in 2014, 15 sometimes. I saw a small team was there. I used the word Tridev. After a few years, when I visited, I used the word, now you are having 5 Pandava with you. You are a Krishna. After that, when I came, sir, I used the word, the 7 stars, Saptarishi Mandal, and you were the polar star, sir. Today, when I'm seeing Swapnil is sitting here, when I'm seeing my very able colleague, Dr. J.J. Patel, sir, is sitting here, Didi Patel is sitting here, I'm encouraged to use the word, the galaxy of scientists who are associated with you. Certainly, this seminar was wonderful seminar, and I'm sure that this seminar is going to be wonderful because it's still 1 more days to go, and I came to know about the brochures you have put out, that that presentations of the tomorrow's speaker is splendid. He is an international speaker. Sir, as I'm speaking here so many times, every time I'm speaking about the microbes only. And when every time I'm speaking about the microbes, today I'm thinking what I should have new for my audience. Sir, basically, I have nothing new, to be very honestly, when I'm sharing this place with Dr. KG Patel, sir, I can't speak wrong. I have nothing new to present to my friends here. The only thing I have the old wine in new bottle. So kindly allow me to share my experience in a very different way today again. Sir, as far as the use of microbes in agriculture is concerned, just now we had a very informative presentations by one of the learned entomologists, Dr. Sikarappa sir. And sir, he has very interestingly and very extensively presented the use of different microbes. I would like to use word microbes, though I spoke only the fungi. Most of the talk was concentrating on the fungi, and soon we will be having another talk by one of the learned scientists from the Jamnagar Agriculture University, Dr. Jetwa. He is also going to speak on the mycoparis, entomopathogenic microbes. So I would not like to touch those things, but he has delivered very good talk on the microbes. But at the end, sir, when he was presenting, sir, I observed that there are so many constraints. When there are so many constraints, then what happens? That oh, your candidate is good, but because of that constraint, when you cannot use your candidate, or when you cannot use your, you cannot exploit your materials, then you have a pain. And to be very honestly, sir, the same pain is there with me also, because we have done a very good work on the microbes and the use of microbes in agriculture for the supplementation of different nutrients being used in plant production. Sir, what we did, that we started from whatever the hurdles are there in the using of these microbes, and then we went ahead. So what are the hurdles and what all these things are there? So I'm going to discuss the most important things what I'm facing, or the most important problem what everybody are facing when they are talking about the microbes, and how this fungi, how these microbes can use it for the crop productions or pest or disease management. Then everybody says, oh, these are the very tiny in the organism, what they can do. But sir, what the lesson we learned from this coronavirus COVID-19 epidemic, that is severe acute respiratory distress syndrome, COV virus, is this, that at least now no one will underestimate the microbes because of their small size, sir. My entire presentation is dedicated to the microbes, and my entire presentation is dedicated to the bacteria for being with me. Why I'm saying that I'm dedicating this presentation to the bacteria for being with me, sir, because I believe in Jainism. And in Jainism, we have been taught that you can live without the food for many days. But if you ask anyone, or if you ask me, can you live without microbes? Probably if we are devoid of the microbes, we can live less than what we can live without the food. I thanks these microbes, these bacteria, for being at a right place. I just want to stress this particular things, that I want to thank this bacteria for being at a right place. Because see what happens, that if the person is not at a right place, and if the things are not at a right place, then it is going to spoil the things, or it is not being used extensively. Why I'm using this word particularly? Because if you see here, one of the bacteria which everybody knows, I would like to start from those bacteria which is known to everyone, is this, that Escherichia coli is the bacteria which is there in the human gut, sir. So this Escherichia coli is a bacteria which is there in the human gut. But if this bacteria comes out of the human gut and goes to urinary tract, then it causes the diseases. So this bacteria, if it is there in the human gut or intestine, over intestine particularly, then it is helping the human being. But when this bacteria comes out of that and goes in another side, so for example, in the urinary tract, then it causes disturbance or causes the diseases, these things. Not only this, sir, this Escherichia coli bacteria is used at many places. So for example, if we have to test the quality of water, then we are testing whether the growth of Escherichia coli is there or not in this water. If the growth of Escherichia coli or the presence of Escherichia coli in this water is there, then this water is not used for the drinking purpose. The most pain which I am having, sir, or the problem which I am having, as Dr. Sekharappa also mentioned here, that we are, I would like to use the word, please pardon me, we are full. Why I'm using the word we are full? Because when we go sometime somewhere and when we select any things, we always pay due attention on this. What we always pay due attention on this in the sense that when we are going in the market to buy a car, we will see we will not buy a car which is having the least features or less features. We will certainly prefer the car which is having more features. When we are going in the market, we are buying the clothes. We are so choosy. When we are getting the shoes, when we are getting ready for the office, and we are changing the shoes every day, that okay, this shoes is not compatible with this dress. When we talk about the cell phone, we discuss with our colleague friends a lot that which phone I should use it. When we talk about the internet connectivity, we discuss a lot about this, that which is having cheaper, which is having good internet connectivity and all these things. Similarly, we discuss this each and everything at each and every place, whether it comes to the employment in the job, whether it comes to the partner, whether it comes to the friend, whether it comes to the fun, like hero, which hero movie you are going to watch it. But sir, the most pain is this, that when it comes to the microbes, we never choose that which microbes are going to be used and which microbe is not going to be used. My concern here is this, sir. Say, for example, in case of the human being, that the different people are having different capabilities, different skill and different attitude. And according to their skill, attitude, and capabilities, different people are selected for the different purposes. But when it comes to the microbes, I've seen many people who are not paying due attention on the selection of the microbes. And the most important thing I would like to communicate here is this, that dear friends, say for example, when we are selecting the Indian cricket team, we are paying due attention, we are looking all the matches, we are seeing how they are performing in the nets, and we are seeing how they are performing in the match to match. Then we are selecting this Indian cricket team. Similarly, dear friends, you need to select the microbes. Especially, I just want to say this to the researcher, that when you are selecting the microbes, you have to select a very good strain of microbes. If you are going to select a very good strain of microbe, then it is working very efficiently. In the beginning, I gave an example of this Escherichia coli. And again, I would like to give the example of the Escherichia coli only. But Escherichia coli, there are so many species of the Escherichia coli. Sorry, there are so many strains of the Escherichia coli is there. But one strain of the Escherichia coli is very pathogenic. If you are having that strain with you, then it will cause more disease. And recently, we have seen in case of the COVID-19 also, we are talk of the town in these days is this, that the new strain of the COVID has entered into the India from the UK, isn't it? What does it mean? It means this, that the different, though this is the species, but we have to further refine it, that the different strains of this, different fungi or different bacteria, whatever you are going to use for the natural resource management is having different capabilities and caliber. Sir, I'm going to highlight the same thing on these presentations in the title, which is known as a microbes, volunteer of par excellence to manage the natural resources in agriculture. I always say, I always mention these things, that microbes are really and the true volunteer with tremendous capability to manage the natural resources in agriculture. When we talk about the natural resources, then a very layman question comes. My son asked me this question, that's why I'm discussing here, though everybody knows here this, that what are the different natural resources? You can see this beautiful lawn here. And from this lawn, you can understand that the natural resources are first land, second air, third water, fourth nutrients, and the fifth sun. if you'll remember it like this as a lawn, it will be easy for all of you to remember that what are the different natural resources. Of course, land is a natural resources, air is a natural resources. God has blessed us with this. Water is natural resources. One of a learned colleague is going to speak today about the water and the water use efficiency. Nutrients, I am going to speak how these nutrients are made, about these natural resources. And the sun, everybody knows, I'm not going to touch it. These are the different services microbes do. Why I call microbes as a volunteer par excellence, because you can see here land also for the land improvement. Microbes are used for water also, for the water quality maintenance. Microbes are used in air also. When we talk about the prevention of air pollution and reduction of carbon dioxide from the atmosphere, or I would say in nutshell, reduction of the greenhouse gases from the atmosphere, then the microbes are used. And the nutrients, I'm going to speak here that how these nutrients are particularly going to improve the soil fertility. Dear friends, before we talk about the nutrients in detail, we have to understand about the interaction of the living organism with the environment. And I'm going to discuss this interaction of the living organism with the environment and this ecosystem work. Dear friends, this ecosystem is nothing, but ecosystem is an area with the uniform environmental conditions associated with the particular species. So what happens that in a uniform environmental conditions, if a particular species is found, then it is making the ecosystem. And in this ecosystem, the human and environment and animals are staying together. It's very interesting to find out, because nowadays everybody is talking about the ecosystem services, not about the ecosystem functions. So we have to understand that what is the difference between ecosystem services and ecosystem function here. Here, friends, many years or many people use this word ecosystem services and ecosystem functions as an interchangeable word, but in fact there is a difference. There is a minor difference between ecosystem services and ecosystem function. If you have to understand what is the difference between ecosystem services and ecosystem function, then you have to understand like this, that every day everybody is going, or many people are going in the morning walk, isn't it? If they are going for the morning walk and again coming to the home and asking their wife to prepare the tea, breakfast and everything, then he is not extending any service to his wife. But what is happening if he is going in the market and bringing vegetables with it, or bringing milk or breakfast with it, then he is serving for his wife. So this is what is the difference between ecosystem services and ecosystem functions. The most important things I would like to hear is this, that the ecosystem functioning is the capability and capacity of the ecosystem to do something that is potentially useful for the human being, for the populations. But when we talk about the ecosystem services, then this ecosystem services include set of the ecosystem functions that are directly linked to the benefit of human well-being. So when we talk about the ecosystem services and ecosystem functioning, we are concerned with the ecosystem services below the ground as well as above the ground. Please pardon me, I'm going to speak about the below the ground ecosystem services only. I'm not going to speak about the above the ground ecosystem services too much, because I'm going to speak about the nutrients. Yes, the time is running out. I will skip few slides. Please pardon me if I'm doing that. But the most important philosophy, what I want to communicate or what I want everybody to understand is this, that dear friends, in ecosystem, this is what we proposed in 2014, sir, that in ecosystem, a balance of good as well as bad microbes are there. If this good and bad microbes balance is ideal, then only this ecosystem is going to sustain, survive, or work in a good way, like ecosystem services is going to provide it to you. And when ecosystem services is provided to you, then you are going to get the good crop. Ultimately, our intention is to get the good crop only, better crop only, economic crop only, without damaging the ecosystem. But what happens when this proportion change, and when the proportion of the bad microbes increases in this population, then what happens? We are facing so many problems in the field when it comes to the crop. So if ecosystem proportion is good, then the good crop is there. when the pathogen proportion increases, the diseases increases, and because of the disease, plant cannot perform well. When the proportion of the insect increases, the insect cause the losses. Dr. Sekharappa sir has discussed a lot about the insect, and Dr. Jetva is going to again speak about this. So I'm skipping these slides, and because of that, I'm skipping few slides more. But it is not like that the biotic stress only. It is the abiotic stress also. And when we talk about the abiotic stress, in particular the nutrients, if there is a deficiency of the nutrients, then it is causing so many deficiency symptoms. But it is not always like this. We have to talk about the balance, not the deficiency and presence of the nutrients, but we have to talk about the balance of nutrients. If you will see this photograph here, this is the watermelon photograph, and the photograph, when I visited one of the field, I observed that the farmer has not used the correct proportion of nutrients required for the growth or for the better yield of watermelon. And because of the disproportion or imbalance in the nutrients, what happened is this, that the watermelon, the entire crop, sir, I visited this field 1 year back, and this farmer was having this watermelon crop in 2-hectare land. And from this 2 hectare, the farmer was expecting to have around 8 to 10 lakh rupees money benefit. He lost it, sir. This is happening in case of the crop. So, my dear friends, the things I would like to discuss here is this, that the nutrients are nothing, but the nutrients are the nature's treasures. Everybody knows about these nutrients, and because of the time constraint, I'm going to skip those slides. When we are having some lacuna, when we are having some missing things, and when we are having some important things, like few of the problem associated with, as Sekharappa sir has discussed, I'm going to discuss only those things is this. What are those missing things here is this, that when we are being taught about the nutrients and its use in agriculture, we have skipped some certain things. Though we take it granted that the student knows this, but what happened that actually the student doesn't know this and then do not use it efficiently. The first and foremost important thing is this. One, we talk about the criteria of essentiality when we talk about the essential nutrient in agriculture. Then you have to remember this: these nutrients would not be an essential nutrient for the plant if this would not be present in the environment. Certainly, because the evolution of plant took place simultaneously in that particular environment. So if anything is present in that environment, then only it is essential for the plant. The another important thing is this: the nutrient requirement by all the living form of life is almost similar to the plants. The only thing is this, that few nutrients might require in certain things extra, few nutrients might require in a higher quantity. But almost if you look at this, the scenario is same. Only primary producers, this is very important things, that only primary producers can assimilate most of the nutrients from the element. I'm writing here most of the nutrients with caution. When we talk about the iodine and iron, this can be assimilated by human being and other animals in like in case of iron for the synthesization of hemoglobin. So it is the, but otherwise in case of this, most of the nutrients are assimilated from the plant only, and then it moves further in the food chain or food web. Then the most important thing, sometimes somewhere we are missing here, that's why we are causing the problems of the nutrient, is this, that the law of conservation of energy and mass is followed in the nutrients and life also. Sir, when I discuss all those points, then the thing comes that if these nutrients are not available in the ecosystem, then why these nutrients are important? Certainly, if these nutrients are available in the ecosystem, then only these nutrients are important for the plant. But sir, what happened? That the God has made one trick, played one trick, I would say. What God has made, or what the God has played one trick, but God has not put those nutrients in a form which is required by the plant. I would like to repeat the same word, please pardon me, that the God has not put or made available those nutrients in the form which is taken or absorbed by the plants. Some people says that God has played the trick. Even I use this word that God has played the trick. But gentlemen, dear friends, God is not here, or God never play trick with anything. The only thing that when we cannot understand the intention of God, we said that God has made the or played the trick with us. But the actual thing is this, that the nutrients are required for the plants, and if these nutrients are available in the form which can easily be assimilated by any one of these, then it would have misused in the ecosystem. When we talk about by taking one simple example here of the wheat, flour, and chapati, when we talk about the life of chapati, it's in few days or hours. But when we talk about the life of the flour, it is in few weeks. I would like to use the word week here. But when we are talking about the grains, the life is of the few years, or I would use month. Year is not most appropriate word here. My concern here is this, that if God would have made the chapati directly, then since it is a perishable thing, it would have perished, it would have spoiled, isn't it? That's why the God has not made the chapati, God has made the wheat. If you are having a skill to convert this wheat into the chapati, you will satisfy your hunger, otherwise you will not satisfy your hunger. But what happened, dear friends, in the early stage, is Dr. Sekharappa sir said that during 1964, at the time of green revolutions, hybrid varieties came which were responsive to the nutrients, which were responsive to the chemicals. And what we did, we thought that, okay, these are the varieties, these are the materials which is responsive to the nutrient and chemical. Then what we did, we could not understand that how to manage these nutrients in the ecosystem. So what we did, we keep on adding the nutrients. Say, for example, when we talk about our agronomical practices also, then we say that, okay, you go for the application of nutrients. Say, for example, I'm giving this rough example. Okay, you add 80 kilogram nitrogen, 40 kilogram P2O5, and 20 kilogram the K2O or potash fertilizer. So what we did, what we have done, we were keep on adding these nutrients into the ecosystem. It was required to keep on adding the system nutrients in the ecosystem, because there is a difference between the forest ecosystem where everything is buried again in the same things, then the agriculture ecosystem, because from the agricultural ecosystem the things are taken away from one place to the another place. Certainly that is required, but still adding the things are not wise in this ecosystem, rather managing the things are required in the ecosystem. Dear friends, the time is very powerful, isn't it? We have seen a big change during this corona COVID-19 situations. The change was this, that earlier when the bullock used to work in the agriculture, we had a strip on bullock, isn't it? And the human was keep laughing. The scenario has changed. Now this has come to the human being, isn't it? That change, we brought it, that's why we are surviving. If we would be adamant at the time of this epidemic is spread at a good pace, that we are not going to change and we are not going to use this mouthpiece, isn't it? This is mouth mask in the sense, then we would have probably not survived. So my question here is this, dear friends, that you have to change it. I'm coming to the direct questions, like direct constant, and I'm going to ask, or I'm going to answer those direct constant only by looking the time frame, sir. If you will permit, I will take another 10 to 15 minutes. If you feel that I'm going a little slow, you can stop me in between. I'm going to skip few slides which I have prepared here, because time is not permitting all of us. We have another 3 more lecture today. So my concern here is this, that how much nitrogen is assimilated from the atmosphere and how much is from the soil. See, this is the important questions. Let me take 2 minutes to explain these things, because this is making a confusion, and a wrong philosophy is there in the scientist, that how much nitrogen is assimilated from the atmosphere and how much nitrogen is assimilated from the soil. This is the questions people ask me, that what these microbes are going to do in the soil when you are going to aid the soil. ultimately the nitrogen is required in higher quantity, and this nitrogen, if you are not going to supplement through the chemical fertilizer, then it is not going to work. So dear friends, to those people, I would like to introduce one of the experiments which was done by the Franco and his co-workers in 2011 from the Brazil. As we all know that the Brazil is the most leading sugarcane producing country in the world. So what they did, they made one experiment. This experiment was very easy experiment, very simple experiment, and very informative, very important experiment which can change the philosophy of different people. That's why I'm going to explain this experiment of Dr. Francisco from the Brazil. What he did, sir, here, we know that in atmosphere 78% nitrogen is there. You cannot change, manipulate that 78% nitrogen which is there in the atmosphere, but we can change or manipulate in a controlled condition the nitrogen which is there in the soil. So he conducted one experiment in the controlled soil in which he added the nitrogen, and that entire nitrogen what he added was a radio label, isn't it? So when he used the radio labeled nitrogen, and then he grew the sugarcane plant. After growing the sugarcane plant at the initial stage, he observed that in the sugarcane plant, 40% nitrogen is radio label, whereas the 60% nitrogen is not radio label. That indicates that the 40% nitrogen has come from the root, whereas the 40% nitrogen has come from 40% nitrogen has come from the root, whereas the 60% nitrogen has come from the atmosphere. That atmosphere nitrogen was not radio label. Then what happened, dear friends, that he observed the same thing at the time of harvesting, that okay, from the sugarcane plant, how much nitrogen is a radio label and how much is not radio label. And you will surprise, dear friends, that only 10% nitrogen was radio label, whereas 90% nitrogen was not radio label. It indicates that in the plant, this radio label nitrogen, which is 10% only, means the only 10% nitrogen is coming from the soil. Remaining 90% nitrogen is coming from the atmosphere. I would like to highlight and mention this thing. In the beginning, when nutrients or criteria of the essentiality was given, and we got the idea about the nutrients, it was said that the carbon is also taken from the soil. But gradually we realized that the carbon is not taken from the soil. Rather, carbon is assimilated in the process known as a photosynthesis from the air. So this is the fundamental and most important activities, where physical energy means sunlight energy, and the chemical energy means carbon dioxide and water is making a biological energy, means the glucose here. So this is the things. A lot of research is going on in these directions. Right now, this is just a preliminary information I just wanted to share. I don't know what will happen to this, but if this research is going to establish for all the crops, then we have to change our philosophy of supplying of the nitrogen. What is the nitrogen cycle? I'm not going to discuss here in detail because everybody knows about the nitrogen cycle. Many people knows about the nitrogen cycle, and they can understand about the things. But the thing is this, that how much pairing capacity of the phosphorus is there, like what is the problem associated with the phosphorus is there. That also I'm going to skip with the simple words this. I'm not going to take much time in these questions also. But the phosphorus is a very funny chemical, very funny nutrients. Why I'm using funny word in the sense that in acidic pH also it is not available, in the alkaline pH also it is not available. It is available in the pH somewhat in between 6.5 to 7.5. Like the maximum phosphorus availability is there between pH 6.5 to 7.5. If the pH is going to be higher, like alkaline pH, then it is going to make the covalent bond with calcium. And if this pH is going to be low, then it is going to make different bonding with the ferrous, aluminium, manganese, and different other cations. So this is the problem with the fire. I don't want to discuss much because of the time constraints. I don't want to discuss this. I just want to discuss about the potash ent. Potash ent is not a very popular name. We have given this potash ent name because if you look like this structure here, you can find that abdomen is there and thorax is there. So like this abdomen and thorax without wing is there in the end. So we have given this potash ant name here. So when we will see this potash ant, we will find that most of the potash is not available or not available in the form which can be utilized by the plant. Like 90 to 98% of the potash is available in the form which cannot be utilized by the plant. Only 1 to 2% potash is available in the exchangeable form, which is utilized by the plant. This is what I am discussing since beginning. And I just want to experimentally prove my words. That's why I'm discussing this experiment here. Do the different strengths have a different capability? As I said, we select the SIM card, we select the mobile, we select the car, but we don't select the microbe. This is what is the problem. When we go and talk, many people nowadays, what is happening? Everybody is saying, you take soil from this, add this, grow microbes yourself. These microbes are going to work. Many people are coming to me in Mercedes car and they are talking like this with me. So we have done a lot of research in these directions. I am not going to discuss about the research in these directions, what preliminary in vitro research we have done. The only thing I would like to share with my audience that biofertilizer production unit of the Navsari Agriculture University is one of the leading unit in the country. And we have produced more than 8 lakh liters of biofertilizers. By looking at the quantum of biofertilizer we have produced, you can make out that how expanded this unit is and what is the level of work we are doing here. So, dear friends, the another important thing what we have done is that we have done all the research right from beginning to the end in the line of biofertilizer, right from the isolation of microbes to the product development. We isolated many strains, and on the basis of in vitro efficiency of those strains, we found that we selected one strain. And after finding one strain, what we did, we compare that one strain in the field also. Very rarely you'll find that kind of research in the field level. You'll find that kind of research generally at the in vitro level. That what we did, we selected one of the best strain of commercial product, one of the most running commercial product in this area, and we selected our strain. Then what we did the experiment without the recommended dose of fertilizer or without any nutrients as well as the recommended dose of the fertilizer. We grow the crop. We did the experiment on the crop, which is known as a finger millet, one of the poor man's crop. But nowadays, because of the nutritional value, the value of this crop is increasing. From poor men, it is going to elite people. So we did research on this, and we surprised that when we are using no fertilizer, the performance of the crop is poor. If you are using the fertilizer, then the crop is giving good growth. I would like to mention here that the crop is giving good growth in the sense that when we talk about the recommendation of fertilizer in this finger millet crop, then this finger millet crop recommendation is very less, like 40% to 40 kilogram nitrogen and 20 kilogram P2O5 only is recommended here for this crop. So in comparison, when we use the commercial based product of the biofertilizers, it was giving a very good result. And our result is it has given in vitro experiment. All we did in the field also it has given the best result. That's why I stress on this, that please you pay attention and take care on selection of the different microbes, isn't it? The another question, very important question is this, that okay, comes that where you can use these fertilizers. Dear friends, these fertilizers or these biofertilizers can be used in wide range of crop, whether they are ornamental, whether they are fruit crop, whether they are vegetables, whether they are cereals, whether they are pulses, like n number of the crops you can use it. This fertilizer can be used in n number of the crop. The only difference is there that for all the crop, phosphate solubilizing bacteria like phosphate providing bacteria and the potash providing bacteria is going to be the same. But for the nitrogen, since you have to put nitrogen in the soil from the atmosphere, like to bring, I generally say 1 inch nitrogen down, you have to do labor. That's why for the legume ecosystem, rhizobium is there. For the cereals, azoospermillum is there, that is associative symbiotic bacteria. Then it comes to the sugarcane ecosystem. Then in case of the sugarcane, acetobacter is there. Then it comes to the other crops, then acetobacter is there. Acetobacter is, dear friends, nothing but it's a free living bacteria. Whereas for the cereals and sugarcane, acetobacter and azoospermillum are associative symbiotic bacteria. Whereas the legumes, rhizobia is used, and this rhizobia are symbiotic bacteria. So this is what is the difference, but you can use this bacteria in the wide range of crop. I'm not going to discuss this data more and more, but we have done a lot of experiment to do this. The important second thing comes that is method of application is also having any significance in the use or efficiency of this biofertilizer. Definitely, dear friends, method of application is also having a high significance in the biofertilizer. See what is happening, that our rhizosphere, the zone around the root, is rich in secondary metabolites secreted by the plants. Around 30% metabolites synthesized or the carbon synthesized by the plant is released in the root system, and our bacteria is utilizing this carbon or these nutrients of the plant. That's why activity of the microbes in the rhizosphere is more than any other places because of the metabolites secreted by the plant. Dear friends, if you are delaying the application of these microbes, then this space is going to be occupied by other microbes which are least effective or no use. That's why early spray, early application, and application of this different microbes along with the seed as well as seedling is having high impact here. And the thing, this is very important questions I just want to discuss here is that what happened that when we talk about the biofertilizers, then everybody knows about the legume and rhizobium, isn't it? Our concept is associated or limited up to legume and rhizobium. See what is happening in case of the legume and rhizobium. The first and foremost important thing is this, that if rhizobium would not have been associated with the legume, then legume would not have been protein-rich vegetables in its ecosystem or in its life. Legumes are protein-rich just because the rhizobium is associated with it. So, dear friends, when this rhizobium is associated with this legume, it is providing nitrogen. But another beautiful things of this crop system is this, my agronomist friends knows better. Okay, my friends knows better that this is going to improve the fertility of soil also. But one experiment we did, whether it is going to improve the fertility of this crop or not, in case of the associative symbiosis or not, then it is there that no, it is not going to improve the fertility of crop in case of the associative symbiotic nature. So we did this experiment, sir. So what we did this experiment. Like the question was this: is it is going to improve the fertility? Like associative symbiotic bacteria are going to improve the fertility of soil or not? To find out this, we did a lot of experiments for the 6 year almost. And if I have to conclude, or if I have to discuss about those 6 year experiment, and from the 6 year experiment, I can conclude like this: that crop performance and yield is increased. If you will compare here, I have not used here, like this is the experiment we did it. I have used here the recommended dose of fertilizers, and the recommended dose of fertilizer when I used it has given the yield as a 124.44 tons per hectare. But when I have used different microbes and have reduced 50%, here the most important thing is this, But in case of sugarcane, recommended dose of nitrogen is 250, 125 P2O5, and 125 kg is K2O. So this is a huge quantity of the nutrients we are providing it. And because we are providing it in the huge quantity, especially when it comes to the potash, then we have to import this potash from the India, because India is not making or synthesizing this potash. We are importing this fertilizer into the country, and then we are distributing it to the different agencies. So to buy this potash, you have to use the foreign currency, isn't it? And always our honorable Prime Minister and everybody in these days are talking about the make in India things and self-dependent, self-reliance. In that way, it is going to help much, because here you can see the two important differences or two important change. One, that we use half of the potash instead of the full quantity of the potash. We are using half quantity of the potash. In spite of this, it is giving more yield, more significant yield. Here you can see the yield is 124.44 kilogram. But if you'll see here, the yield is 133.56 kilogram. So yield is also significantly higher than this. And the second thing is this, that the half of the chemical fertilizer is used here, and we are using half of the chemical fertilizer, and at the same time, and we are increasing the yield of the crop. Then the dream of our honorable Prime Minister, which is doubling the farmers income, can be realized. So dear friends, my concern here is this, that if you are using this biofertilizer, so the bacteria which can be used as a biofertilizer to realize the higher crop yield, and to use it, you can help farmers in doubling his income, you can save the foreign currency, and at the same time, the most important things we need to care is the ecosystem. Even if we have to pay higher amount of, or even if we have to increase higher cost of production, we don't mind if we are saving the precious ecosystem. Here, important thing is this, the same treatment we have used here, this treatment 9, what we have discussed, where half doses of the different chemical fertilizers and biofertilizers were used here. The most important things when we talk about this, that EC and pH is a one thing which is not much changed because of the biofertilizer. The reason why it is not much changed because of the biofertilizer is this, that already it is in a proper way. The second thing is this, that what is happening in case of the nitrogenous fertilizer in the soil. This is what I just wanted to highlight here, that in this case, the fertility of the soil has not improved as much as we are observing in the legume ecosystem, because when we are using the legume, then soil fertility is increasing. And in the different crop system, when any crop is sown after the legume crop, then in that circumstances, what is happening? The residues nitrogen, which is available in the soil, can be utilized by the other crop. That's why in the sequence, legume and the cereals are grown. Here, in case we have to pay this attention, that the soil fertility has not improved that much. We worked on this, that why the soil fertility is not improving. We did experiment for around 6 years, and then we concluded like this, that why the soil fertility is not increasing here. Because in case of the legume, what is happening, that the bacteria are associated with the root. So when the bacteria are associated with the root, and after harvesting of the crop, everything remains downside in the root only. So it is fixing the nitrogen and it is giving it to the soil. But in case of the sugarcane, it is the associated symbiotic bacteria, and more number of the bacteria are there in the internodal part in the sugarcane crop where ibud is coming out. And because of its presence in the internodal part, it is not fixing and not improving the soil fertility. I just wanted to discuss these things, because what happens that when we talk about the biofertilizer, then what people do, that okay, I have added biofertilizer in one crop season, so I need not to add the biofertilizer in another crop season. But that is not happening, my dear friends. you have to add those biofertilizer again and again in the crop ecosystem. This is what is an important message I would like to communicate to the scientific fraternity. Then what happened there in this different ecosystem, that the P2O5 reduced, K2 also reduced. Why this K2 and P2O5 has been reduced? Because in general, what happened, that the crop is using higher quantity of the potash than what we are providing through the chemical fertilizers. Even whatever the residue available in the soil is also used apart from the fertilizer what we are giving here. But what happened in case of the bacteria when we are using, here you can see that the sharp increase in the level of phosphorus and the level of potash is there. See why it is happening? It is happening just because that the bacteria which we are using, phosphate solubilizing bacteria and potash mobilizing bacteria, this phosphate solubilizing bacteria and potash mobilizing bacteria is growing and multiplying in the root system. When they are growing and multiplying in the root system, they are secreting certain organic substances or certain organic these acids. Those organic acid is going to convert the different phosphorus which is available in the soil but not available in the form of available potash phosphorus, and the same thing with the potash, it is going to convert from the unavailable form to the available form. That's why here we can see the improvement in the soil fertility. This is what the important research, dear friends. We did it for around 6 years because we were not convinced with the information what we obtained in the 1 year observation. So we keep on doing the research, and then later on we have tried to find out where the bacteria are colonizing and in how much quantity this bacteria are colonizing. After this rigorous research, we came to know about these things. The another important questions which is coming in any innocent mind, especially when we are selling or when we are distributing this biofertilizer to the farmers, and the question is like this, that are these bacteria compatible with chemical fertilizer or with the other chemicals? See, one common question is being asked by all the users here, and one common philosophy is there among all the literature and all the people who practically has not worked extensively on different microorganisms. You will find many people recommending like this, that okay, if you're using the biofertilizers for any microbes, then you should have a gap of certain days between the chemicals, isn't it? Chemical means chemical fertilizer here. That's why what is happening, that as the basal fertilizer is applied, isn't it? That's why many people say you don't use biofertilizer because you have applied the fertilizer here as a basal dose, and the fertilizers and chemical fertilizers and biofertilizer cannot be used simultaneously. The second thing also about the insecticide, about the weedicide, because in these days, yesterday Dr. Tushar has discussed it very well, that what we did it, we were more depending upon chemicals, isn't it? And our intention was earlier to remove the weed from the ecosystem. But nowadays we are discussing about the management of weed. So we have to take care of these kind of informations, and this type of the points here is this, that we don't want to remove the weed from the ecosystem. But earlier we were removing the weed from the ecosystem. I'm talking about the incidents, what is happening when we are planning to remove the weed from the ecosystem by using chemicals. So, there are so many chemicals we are using as a weedicide. Dr. Dikshit yesterday has explained it in a very informative talk. So I don't want to discuss about all those chemicals which is used for the management of weed, but ultimately we are using this. Are our biofertilizer are compatible with those materials or not? Because ultimately the weedicides like pendimethalin, 2,4-D, whatever, pendimethalin and all is going to be incorporated into the soil ecosystem, and the biofertilizer is also going in the soil. So these are the compatible? Dear friends, we did a lot of experiment here to find out the compatibility of different chemicals with our isolated microbes. To your surprise, to my surprise, when I planned this experiment, sir, I was under the opinion, though I was having good knowledge of the biofertilizers, I was having good knowledge of the plant pathology and good knowledge of the different chemicals. I was under the impression that this chemical are going to at least reduce the growth of those microorganisms. But I was surprised when I came to know that our microbes are smart enough to even utilize some of the metabolites from the recently developed chemical and even perform better in the recently developed chemical than the normal conditions. I did this experiment as many as 5 times to confirm these things, and then came to know that bacteria are used in the biofertilizers and bacteria are not killed by the fungicide. Bacteria are not killed by the insecticide. Bacteria are not killed by the weedicide. And the bacteria are not killed by the nematicide we tested. I don't want to give here the name of those nematicide, insecticide, and fungicide because those are very commonly used insecticide, fungicide, and nematicide. But whatever the fungicide, insecticide, nematicides we tested, our bacteria were not killed by this. And then finally conclude that, dear friends, the bacteria is killed by the antibiotics only. A special group of chemicals are there to kill those bacteria, and those are the antibiotics. So if you are using those antibiotics here in this system, then only, dear friends, this bacteria can be killed. And we did one more important experiment here is this, that what happened when we are adding this antibiotic in the soil. This experiment is still going on. I just want to discuss the preliminary result with my colleagues that, okay, if this bacteria can be killed by the antibiotic, and if died with the accidentally or by for some other purposes, farmer is mixing those antibiotics in the soil, then what is going to be happen? Dear friends, I am working with the initial antibiotic which is used, especially the tetracycline and penicillin. Then only I will come to the higher antibiotics or fourth generation of the antibiotic which is being used in these days for the medical purposes only. But the thing is this, I'm finding a rapid degradation of those antibiotics in the soil. I don't have much result to share with all of you, but I just want to say to my audience and my colleagues that we are working in that direction, that how the antibiotic is going to change the soil microbial ecosystem. Our title of research or interest is in these days is this. And so the metagenomic study, we are going to plan this study. Everybody knows that this is the era of metagenomics, and we are switching over to the metagenomic. I would like to conclude with these words that there are so many hurdles we are facing. There are so many hurdles. There are so many negativity in the field about the use of fertilizers. But with the support of our authority and with the support of farmers of the South Gujarat, we were in the position, we are in the position to commercialize these biofertilizers, and we have produced more than 8 lakh liters of biofertilizer by working on the different hurdles or the problems which Dr. Sekharappa sir has discussed, and we are successful in commercializing this product. We are happy to announce this, that this product is marketed or sold by the Navsari Agriculture University without advertisement. Means only the mouth publicity by the farmer is there, and because of this product is being marketed. This Navsari Agriculture University and this biofertilizer production unit of Navsari Agriculture University is pioneering in making the capsule formation of biofertilizers. Whatever the capsule formation in these days we are listening from the different people is developed after getting inspiration from the work done by the Navsari Agriculture University. You can see our Honorable Prime Minister's photograph here, holding 2 capsules in his hand. This is the photograph of our Honorable Prime Minister when he was our Honorable Chief Minister of Gujarat State. Everybody knows that more than 7 years, he has more than 8, around 7 years, yeah, he is there as a Prime Minister. And this is the photograph of even before that, 2 to 3 years before that. So like my concern here is this, that we have produced that materials. You can see the similar kind of capsule sitting here and the similar type of capsule in our Honorable Prime Minister sir's hand. So Navsari Agriculture University is the first university or first institute in the world which has produced the capsule formations, and our future plan is also like this. I would like to end this my presentation with one word like this, that this is the dream of our Honorable Prime Minister Sri Narendra Modi ji when he was here as a Chief Minister of Gujarat State. He said one word. I just want to repeat his word. He used that word in a Gujarati, but for better understanding, I will translate that word into the
Dr. Lalit Mahatma, sir, your screen is freeze. I think he might have faced the issues of internet connectivity. Dr. Lalit Mahatma, sir. Dr. Lalit Mahatma, sir. Are you there? Okay, anyway, participants. Is there, you have any questions regarding the presentations of Dr. Lalit Mahatma, sir? You can put your questions in chat box. Even you can raise. Yeah, I observed one questions. I think he might have faced connectivity issue. So without delay, I invite Dr. Jaimin Pandya, as we know that sir deliver very comprehensive and very resourceful lectures on the very emerging topic, that is on microbes. And I think it is not only resourceful, it is also very thought-provoking lectures. Of course, yes, Dr. Lalit Mahatma sir is there. So just soon, he will soon join the webinars. Dr. Lalit Mahatma, sir. I request Dr. Swapnil Deshmukh kindly, Dr. Lalit Mahatma sir is here, so make him a co-host so he can share their view.
Yes, sir. He will co-host and he can.
Thank you very much, sir. I was at the end of my presentation when there was a poor connectivity or loss of connectivity. I have discussed whatever I wanted to discuss. Without wasting much time, you can continue the next session, please.
Sir, one query in your chat box. Please check your chat box.
So there is one query in the chat box.
No, actually, sir is continue his presentations, I think. Sir is trying to share his screen.
Sir, it is almost done when it was closed. I was at the conclusion, sir. So, conclusion this, that you should use biofertilizer. It is having good efficiency, good. I'm unable to see my chat box.
There is one query. I will read it for you. Ramesh is asking, what is the current status of encapsulated biofertilizers and how these are different from the liquid form? Which one?
When we talk about the encapsulation and the capsule formation, both are different things, isn't it? It is not an encapsulation of biofertilizer, it is a capsule form of biofertilizers. So in capsule form, we have done research on this, and right now we are standardizing the mass multiplication protocol. But our concern is this, that we should have a commercially viable mass multiplication protocol, isn't it? So research, as far as research is concerned, we have done the research on this. We have one recommendation out of that in 2018-19, isn't it? But we are working on so that we can provide a cheaper material to the farmer. Second thing is this, that the fertilizers, whatever when we have to use according to the fertilizer control order, isn't it? So we need to have a better idea about the fertilizer control order, whether it can allow us to market it in the market or not. So this is what is going on.
Okay, thank you very much, sir. First of all, I am thankful to the Dr. Tushar Patel who inviting me to give a vote of thanks to the speaker who always inspiring me and feeling proud me, or we can say feeling proud of us, as I was one of the student of you, sir. Besides my academic and research activities, I know you very well, sir, and I have worked with you also, sir. So it's my immense pleasure to give a vote of thanks to you, sir. Sir, you have very well presented your presentation. As I know your skill, very extraordinary presentations you have, so many. And of course, you have so many food stock to serve us also. But time is the limit, sir, actually. Okay, sir. And after all, no words I have to, I have no words about your presentation and for you, sir. Because DBS sir is told me like that you have to return each and everything, and you have to point it out each and every point. But sir, I know you very well, and you are very well presenter, you are very well writer, you are very well speaker, that all things within you, sir. So thank you very much, sir, for being with us, and thanks a lot, sir. Thank you.
Thank you very much, the entire team, for inviting me and giving me an opportunity to discuss the August gathering. Thank you very much.
Again, thank you so much, sir, for your comprehensive, resourceful presentations. Rather, I said it is also thought-provoking, sir.
Thank you very much. Thank you very much.
Now I invite Swapnil Deshmukh to invite our next resource persons, please.
Yes, thank you, sir. Now our next speaker is Dr. D.M. Jetwa, sir. He is working as an associate research scientist at Department of Entomology, College of Agriculture, Junagadh Agriculture University, Junagadh. He is in charge of Biocontrol Research Laboratory, and he has also been awarded as a best teacher during 2013-14 from Junagadh Agriculture University. He has guided more than 2 students of PhD, 12 students of MSc, and presently he is guiding 7 students for the doctorate degree and for 2 students for the master's degree. He is engaged in the course teaching also, and in the experimental learning programs also at UG level. In case of research, he has handled lots of other agency projects and very national level projects like RKY, Horticulture Mission, and ICR projects, and by which he has established this biocontrol research laboratory at Junagadh University. Then he has also registered 3 Indian patents. It is very big, this achievement, that he has registered 3 Indian patents. That first is functional trap for managing insect pests. Then MDP for pink bollworm in cotton, and MDP for brinjal shoot and fruit borer. He has also contributed 32 recommendations for the farming and scientific community. And in case of extension activities, he has conducted a number of trainings for the farmers and delivered more than 100 lectures and 15 radio talks for the farming community. So we welcome you, sir, and thank you for sparing time for us. Now the platform is yours, sir. Dr. Jetwa, sir. You can unmute yourself.
Okay, thank you very much, Dr. Saib, for giving me chance to present in this, national level seminar. So here, as the, I am audible.
Yes, sir. You are audible.
Yes, yes.
I
Please
have
share your screen, sir.
Have you seen my screen?
No, sir. Your screen is not visible. Please share your screen.
Okay, I will try. But now?
Yes, sir. It is visible.
Oh. Okay. Can I start my presentation?
Yes, sir. Yes, sir. Please, sir.
Okay. So
sir,
here,
your voice is somewhat low.
Okay. Voice is low. Now you audible?
Yes, sir. You are audible.
Okay. So here the topic that given me is the role of entomopathogenic fungi in entomopathogen indicated pest management. So here, as the earlier two lecture I already given on the entomopathogens, as well as on the this Mahatma also given a lectures on pathological point of view. So I I am trying to justify myself regarding this entomopathogen. So we all know that the microorganism play a major role in numerous metabolic processes like the nutrient cycling, the environment detoxification, as well as decomposition, oxygen production, as well as so in that way the microorganism or microbes play a vital role in the for the management of many insect pests. I am only concentrated with with the management of insect pests and omitting this pathological point of view. So here we all know that the more than 3,000
Sir, your voice is very low.
more than 3,000 in that we all know that initially as the honeybee and the silkworm were the first arthropods that we were observed and recorded the suffer from many disease by the some fungi like the Bevaria or Metarhizium. As earlier in the history, when we see that the Agestobacci excellent contribution regarding the establishment of the relation of the microorganism like the Bevaria bassiana, this white muscadian fungus with the silkworm. So this is the also one of the important in the uh, in the science of the entomology. So then after Matt Koenig also published a very good work on the green muscadian fungus Metarhizium anisopli, and then the Steenhorst have a major giving a major role regarding the work on these entomopathogens. Now, when we see the consumption point of view, as we all know that the India is the largest producer of the pesticides in South Asia and second largest manufacturing of the pesticide chemicals. So in that way, the 4% of the world crop area and 1.7% of the world pesticide consumption, you can see that in the slide there is a crop wise I have given. So in that way, the major for the management of any insect pest, the major role is the chemical pesticide. Now we all know that when we are using the chemical pesticides, so what the chemical pesticide doing? It's not all pesticide, but most of the chemical pesticides are either we can say that on human being as well as on the agriculture environment there is a major impact, negative impact. Like the in a health point of view, we can say that the is directly many pesticide directly affect on the nervous system as well as initially it is either in the it affect on the irritating the skin or eyes. Sometimes pesticide also a carcinogenic. Many pesticide available in market is a very carcinogenic. It directly affect on the hormones or endocrine system. Sometimes birth defect also we have seen. So in that way, pesticide doing very brusque and it need to stepwise reducing the pesticide and moving to the organic farming, or we can say that using the entomopathogen for the management of insects. So they can we can nullifying the yield point of view. So in that way, why should we concern about the using the pesticide for the agriculture point of view? So these are the some of the most important point that we have to consider, like the toxicity point of view, as it directly toxic to the some of the important beneficial insects like the our predators, parasite, entomopathogens that uh, classical in classical biological control. It's the most important point of view that these are the bioagents that kill by this pesticide. Even when we are spraying continuously same pesticide, at that time resistance problem also we have seen. So insect resurgence we have seen, biomagnification, as earlier Mahatma told that the biomagnification also we have major problem. So in that way there is also a pollution of the air, water, in even soil we have polluted day by day. Many pesticide like the already banned DDT. You can see that DDT half life is more than 14 years. So in that way, the pesticide doing very worse for us, and we need to remove stepwise so that it directly not affect on our yield. So in that way, what is the alternative? So what are the alternative available for the instead of pesticide we can use? The most important alternative that available nowadays is these biocontrol agents. Biocontrol agents mostly includes the predators, parasites, and this entomopathogen for the management of insects. So this is the main point of view. These are three components that in biological control. Here we are only talking about the entomopathogens. As we all know that in the insects below the economic injury level. So here there are a number of entomopathogens are available that now commercially available, from which the fungi, bacteria, virus, nematode, protozoa, and rickettsia are the most important pathogens. From these six pathogens, when we talk about the commercial point of view, the fungi, bacteria, virus, and nematodes nowadays available in India. When we see in the nature, the virus, bacteria, fungi, and protozoa are mostly performed to regulation of the many insect pests as well as mites for the management of insects. So here when we categorize this as a fungi, bacteria, virus, it is a mostly ingested microbes as well as a penetrating microbe. When ingested microbes, we are talking about these type of group which includes bacteria, protozoa, like virus, must be ingested into our body through the infection. and can be considered similar to chemical insecticide like the stomach poison. So these type of bacteria and all need go through our mouth or through the foods for the insects, and it multiply in the digestive system and it kill the insects. So this is the one type of ingested microbes. Here in when we talking about the bacteria mostly cause spreading the after going into the stomach, mostly difficult in the tissue and body fluids. And some bacteria kill the host purely by the activity of the toxin. Mostly we can see talking about these BT, Bacillus thuringiensis bacteria, so delta endotoxin that develop. So it's the same type of toxin developed by the bacteria and it kills the insects. So this is the one main type of groups are available in this all pathogen. One another type of groups are available that directly enter through the host through the integuments. Mean the integument or maybe spiracles, through spiracles of the insects, they directly, or maybe in simple word we can say that through their body or through their cuticle, they directly go into that. And that in that type of group we include these mostly fungi. Fungi is the one of that group that we have include that for the management of the insects. Now for the successful utilization of the these bioagents, or we can say these entomopathogen in the field, the most important things that we have to consider is this the host insects as well as a pathogenic microorganism biology as well as characteristics. Most important things for this the biology as well as characteristics of the insects. Now for the successful
Dr. Jetwa, sir, is there any problem, sir? Okay, I think there is some technical problem. We will make it over in 5 minutes. Please bear with us, all participants. Okay, the issue has been sorted out. I think Dr. Jetwa sir is with us again. Jetwa sir, please continue.
Sir, first you unmute yourself.
okay, can I audible?
Yes, you're audible.
Okay, thank you very much. there is an internet problem issue is going on in Junagadh. So that's why sorry for that. For using any microorganism, three major points are most important. One is the host insects. Host insects. Another is the pathogenic microorganism that we are applying, and another is a
Dear Dr. DM Jethwa Sahab, you are not audible. Dr. Jethwa Sahab, you are not audible.
Participants are requested to bear with us. We will shortly retrieve Dr. Jethwa sir. I think he might have faced very bad internet connectivity.
hello. Hello.
Yes, sir. Yes, sir. You are audible.
So here a major problem is going on internet. So that's why continuously interpreting. So
Yes.
we are talking about the successful use of any microorganism. The three most important factors are
Dr. DM Jethwa sir, your voice is very low.
now available. Hello.
Hello?
Hello.
Yes, sir, you are audible.
Okay. Okay.
Okay.
So this is the main three factors that important is the host insects, pathogenic microorganism, and environment for the for the use of any microorganism in the field. So here the most important thing is that the host insects, their biology as well as pathogenic microorganism, their biology and their characteristics are important for the application in the field. And another thing is that the when we applying any bioagents like any microorganism in the field, the most important is the like their humidity and temperature is the most important for the multiplication either in the maybe in the soil or maybe in the field. So here when we applying any bioagents when we see the status of entomopathogen in India, as per the Central Insecticide Board and the Registration Committee, there is a number of biopesticides that registered in India. And you can see that the more than 15 microbial products, more than 15 microbial species and 970 microbial formulation at that time registered in Central Insecticide Board. As well as when we talking about this Bacillus thuringiensis, more than 30 products that already registered in the CIB. And virus point of view, more than 22 products of two viruses like the Helicobacter armigera and Sporopera and PVR already registered when we talking about the entomopathogenic in marketing in entomopathogenic fungi is we all know that the fungi that causing the disease, the fungi that causing the disease in insects, it is a called entomopathogenic fungi. Mostly these all and filamentous branch structure as well as in layer. So this is the all fungi that available in market, or we can say that in India. From this, most important fungi that are the Beauveria bassiana, Verticillium lecani. Now the name is changed, Lecanicillium lecani, Metarhizium anisopli, then Numeria riley. The name is again changed. It is a Metarhizium riley. Then recently we all know that the coconut spiling whitefly is the major problem in Gujarat. Now in 2016 it is a problem in South India, and now we have a major problem in our coastal belt, either maybe in the Navsari or any places or in our Saurashtra region. So in that way, one another fungi, particularly for the white fly point of view, Escherichia fumosorosia is the most important fungi for this white fly. So in that way, there is a number of fungi is available. Another is the Hirsutella, also a very good promising fungi that are very effective against many mites, even these idiophyte mites. So this is the many registered fungi that available in our India. Now, as this is the common thing is the mode of action of this all fungi. Mostly these fungi are penetrated through the integuments, and some another entry like the spiracle, guts, and elementary tracts also reported by the many scientists. When the favorable condition like the temperature around 30 to 35 degree and around the more than 60% humidities are available, during that time these fungi their Canadia germinate into the short germ tube, and from that one swelling is appear, and this swelling is called apressoria, and from this apressoria mostly attached to the cuticle of the insects, and from that after that there is a one infective peg is developed, and from that peg mostly through enzymatic activity this cuticle go into this cuticle and inside the multiplying the fungi. Now when after the crossing this cuticle barrier, the fungi mostly move to their hemocoel and internal organ of that. Then after their fungi fight against the cellular defense reaction. And if there is a two possibility, if the insects is overcoming this fungi, then normal development of the insects are developed. And if the mycelium maybe overcome this barrier, then it invade the insects and kill the insects. So these the two possibility are there. From that, there is a chlamydospore is developed, and from this chlamydospore, there is a spore are developed. So in that way, this is the process of multiplication of these all entomopathogenics. There are during that time there are number of toxins are produced, and nowadays for the future point of view, these toxins are most important. For example, some of the toxin name I have already wrote that. So for Beauveria, it is a Beauveriacin, Metarhizium is a destruction, and to this is the toxins are developed. Now for the genetical point of view, if we extract this And from that, we have identified some of the places, some of the sequence area that particularly important for the this production of these toxins. Now in the future, we are trying to this multiplying this on this E. coli bacteria for the multiplication of these Bavaria's in toxin. And insecticide just like a available. So these are the common symptoms. I'm quickly moving up that. This is the after the infection of the Bavaria. You can see the in photograph. Initially, these spores are developed on outside. So during that time, it may be if the fungi is the Bavaria bassiana, it is a whitish color spore are developed. If the fungi is a Numeria riley or Metarhizium riley, then it is a pelletage green type or coffee growth we have seen. If the fungi is a Verticillium lecani or Lecanicillium lecani, there is a some coffee growth of whitish color we have seen on that. So in later stage I will show you that. So first Bavaria bassiana. We all know that it is a white muscardian fungus. When we put on the petri dish here, this is the photograph of that. We can see that the whitish color, pale white color colony we have seen. We have isolated many strain of the Bavaria bassiana from the as well as in the pink bollworm, white grub, many other sucking pests from that we have isolated strain. From the pink bollworm point of view, we are the first from India that we have isolated the Bavaria bassiana from the pink bollworm. So this is the white muscardian fungus. This fungi is a very promising fungi as it is a not only effect on the lepidopteran only on the larva, but it's a sucking pest also a very giving a very good result. So in that point of view, this is the most important fungi from all fungi that available in the market. So this is the host range. You can see that each and every larva is infected by this host. You can see the species wise. This is a number of you can see the either potato weevil or unicorn beetles, and even in the sting bugs and cicada. There are number of even in the wasp, even in the honeybee. We have also noticed this thing, but mostly honeybee is a escape because it is a very fluent flyer. So mostly Bavaria escape from that. So this is the one or two sample that we have collected from last 10 years. So in that point of view, it is not much to worry about that. Otherwise, most of the insects that are found on the crops are infected by this Bavaria bassiana. So these are the some of the important market available products that available in India. So same way, one another is another fungi that now is a Metarhizium anisopli. Metarhizium anisopli also very promising fungi. Initially, it is called Entomoptera anisopli. It's a very good fungi, and mostly that we have isolated this fungi from the white grub. White grub is we have a major problem in groundnut in our area. So in that perspective, we have a number of strain of this Metarhizium anisopli. Two to three strain from the white grub and one from the mango hopper. Mango hopper strain is a very good strain that from mango apotek pocket, and it's a very good giving a very good result. And we have for the demonstration purpose we have given to this farming community. So many. So this is the Metarhizium anisopli. You can Metarhizium anisopli. Here is the most important thing is that actually it is a greenish color fungi, but their intensity, or we can say that the coloration is changing by initially it is a whitish color, and then after when trace is given, then it is a greenish dark color. So it is a typical fungi that we have seen in the field as well as we have multiplied in the laboratory. So this is the some of the photograph of that. You can see that in bug white color. So this is the commercial products that available in the market. This is the then this the all products are available in the market. Same way, another is a Numeria Riley, or now it is a called a Metarhizium Riley. It is a typical pelletized green color, and when we this type of fungi Metarhizium we have isolated from our area from the Heliothes as well as in Spodoptera. This is the we can say that this is the perfect example of classical biological control. So in our area, when we going in the field, either maybe in the university area or maybe outskirts of the university, we have seen a number of sample we have collected of this Metarhizium riley or Numeria riley from the field, and it is the most promising fungi for the management of many insect pests. So these type of fungi mostly mode of action are similar to the earlier the Beveria and Metarhizium. So I am moving up that. This is the you can see the then move to the pelletized green color, pale green colors we have seen that. Now this is the old then. Now another fungi is the Lecanicilium lecani. This is the most whitest hollow fungi. You can see in the photograph also. One of them we have isolated from the aphid. So from the aphid, this is the most important promising fungi Lecanicilium lecani, or earlier it is a called Verticillium lecani. So this fungi we have isolated the from the field. This is the you can see that the colony as well as sporulation. So this is the commercially available fungi in the market. So mass production point of view, here the most important thing is the for any entomopathogenic fungi, there is a two way are available. One is the liquid fermentation techniques are available. One another is the solid fermentation techniques are available. So nowadays we are using a fermenter for the liquid fermentation techniques. So it is the commercially point of view, it is a very easy to multiply in the field. Here in the liquid fermentation techniques, there are fermenters are available, and these fermenter mostly we are controlling the temperature as well as their O2 level and all their different protocols there are available, and through which we are multiplying these two liquid fermentation techniques. And traditionally solid fermentation techniques are common. This the given this we have the solid fermentation technique doing at that time in the India, and we are also doing this solid fermentation techniques mostly on the sorghum grain. So in the economic point of view, this is the also a cheaper. And initial if anyone want to start the business of biopesticide, this is the simplest way we can easily multiply these all entomopathic fungi in this. So this is the products. Well, lower side you see that the number of fungi we have registered in this NCBI portal. These are all are this JUBP2 is the one strain is that we have isolated from the pink bollworm. Another is from the Heliothes, one from the thrips. That number of strain, more than 14 strain we have registered in this portal. And this is the our products that now we are going to the commercialization, and we have registered also in the CIB for that, for in the within a 1 year maybe it is a register. So this the things are there. So you so from the last this is under RKVI project, and one another project is a horticulture mission, through which this biocontrol research laboratory we have developed. And through as per the guideline given in under the RKVI projects, and our students like under the financial learning program also we have given the training to this all the undergraduate 8 semester students to for the production this biopesticide. So under that last you can see that from the 12 13 we have more than 5 lakh kg of Beveria bassiana and 14,000 kg of this Metarhizium anisopli given to the farmer as well as for the research purpose to the scientist as well as for the field experiment we have given. So this is the data of that. Now we are moving to the one another is the virus, entomopathogenic virus. So there are a number of viruses are available in the market, but there are when we are using any virus, there is a number of constraints also available. When we are using virus, we all know that the for the virus point of view require living host for the multiplication of this virus. So that's why these type of entomopathogenic virus is now is not available mostly in India. There are more than 3 or 4 companies, including this biocontrol research laboratory of our Junagadh, is this developing and giving to the farming community on no profit or no loss basis. Otherwise, this virus for commercially point of view, it is not most important. If there is any now in future, if any techniques are developed, then we can easily multiply this virus in the laboratory. Then it may be in future is a commercialize the things. So this is the simple things are there. Virus are available. Mostly it is a inclusive virus and non-inclusive. Mostly at that time in market available is a polyhydrosis virus are available, like the Heliothis nuclear polyhydrosis virus, Spodoptera nuclear polyhydrosis virus. One another we have isolated this Spodoptera frugiperda. Spodoptera frugiperda, you all know that last 3 years it is a major pest of the maize. So for the management of this Spodoptera frugiperda, we have isolated this nuclear polyhydrosis virus from that, and we have multiply in the laboratory, and now we have given to the farming community for the demonstration purpose in around our area. So these are some of the families that most important for the virus point of view, from which the nuclear polyhydrosis virus as well as these cytoplasmic polyhydrosis virus are the most important for from that. So it is a mostly this nuclear polyhydrosis virus are mostly it is a rod shaped virus around 20 to 15 micro diameter is there. It is enclosing two proteinous membranes are there, and which together with the particle constitute the polyhedral body or polyhedra. Now one or more virus particle are mostly found in the polyhedra, and this polyhedra is usually hexagonal structure. We can see on the 100x on the microscopic, we can such a DNA are in structure, while the cytoplasmic polyhydrosis virus have a RNA in their structure are there. So as I earlier told that this the virus are mostly go through is stomach poison are there. So mostly through their food, virus are mostly going through their body. If there is techniques are available in market for easily multiply commercially, then this is the promising nuclear polyhydrosis pipe for the management of the either maybe this easily multiply or Podoptera frugiperda. Otherwise, it's when we need a host to multiply. For example, if we want to multiply Heliotis nuclear polyhydrosis virus, we need a Heliotis for multiplication. So in that way, commercially point of view, it is a slightly difficult. Otherwise, it's a very promising nuclear polyhydrosis virus for the management of any insect. So this is the mode of action. Mode of action are mostly virus have a similar, so that's why I'm moving faster. So this is the some of the important things are that the infective larva mostly will eat normally for couple of days, and after 48 hours, if the larva is smaller, then mortality rate is more, and larva is bigger, then mortality rate is less. So this the vice versa things are there. After that, the body becomes a fluid and skin are ruptured, and mostly larva have a oxygen less. That's why it's moving upward upper side and hanging towards the lower side. So this is the main symptom that we have noticed when we are dealing with the nuclear polyhydrosis virus. When the dead larva become hanging from the laying, that the cuticle may be very fragile and left. And when we touch any cuticle, it's maybe very fragile, and we can see that some whitish color liquid is coming out from the larva. So from that, we can easily identify that this is the nuclear polyhydrosis virus. So this is the mass production. Mass production, I earlier told that for production of any nuclear polyhydrosis virus, we have to multiply their host. For example, if we want to multiply Heliothis nuclear polyhydrosis virus, then Heliothis we have to multiply in the laboratory. Now, after when it move to the fourth and fifth instar, then we we exposed to the virus, and in that way we extracted virus from the pit. In that there is a one good recommendation we have done. We have developed our
Jetwa sir, you are not audible. There is again network connectivity problem. Dr. Jetwa sir. So we will wait for 5 minutes. Please bear with us. Dr. DM Jetwa sir.
Can I audible?
Yes, yeah, you are audible.
Okay. So sorry for continuously there is a problem of internet here in Nagar. So sorry. So I am quickly moving to the slide, moving like so that there is a time limit is there. So there are a number of one another is a entomopathogenic bacteria is there. So bacteria is also very promising. Nowadays we all know that the bacteria bacteria for the BT when is coming at that in that point of view the Bacillus thuringiensis is the most promising bacteria for the management of any insect, and there is a toxin are available, and from that toxin we can directly, as we all know that BT cotton is available, so that toxin, so in that point of view also a BT is the most important for the management of any insects. So this is the mode of action is there. there is a symptoms are there. So I am quickly moving that due to lack of timing. So there's a number of commercial products are available in India. You can see that number of varieties are available. Israel is in Karak Shakti and all that. So one another is the entomopathogenic nematodes are there. Entomopathogenic nematode is the most important nowadays because for the management of a white grub in groundnut, entomopathogenic nematodes are most important for the in that point of view. So this is the mostly the stylus less nematode, so it is not injured to the our crop. So in terms of pathogenic nematode, in that there is a number myrmidioides nematode, stenonematode, and heterotalbiditis nematode. These are the three most important family for the management of any insect. So in that point of view, here stenonemma is there, heterotalbiditis nematodes are there. It is a very promising result against white grub in groundnut. So in that point of view, it is a we have isolated one heterotalbiditis indica from the field, and now we are moving to the commercialization of this heterotalbiditis in the market. So it is a very promising nematode, and now there is a number of scopes are moving due to time lapse are there. So this is we all know that there is a pest resurgence are there. So we all know that these two species nowadays is available in India. So for the commercial point of view, so this is the mode of action of this EPN. We can see that EPN are mostly to the anus or pseudopodia or spiracle. So with the mostly is going inside the insect's body and multiply there. So there is a number of another
I think Dr. Jetwa sir is still facing the serious problem of internet connectivity. So I request Dr. D.D. Patel sir to take.
Okay, I think Dr. Jetwa sir is facing the internet connectivity problem. Uh, uh, show
Surely, he will join soon. But I think we should provide only concluding time to Dr. Jetwa sir.
sorry for
okay, Dr. D.M. Jetwa sir.
So I am moving to the.
Please going to directly to the. You have done a very excellent job. So please, you are also facing the internet problem. So go to the. We are requesting you to go. Okay. Thank you. Concluding remarks, sir.
So in that way, the entomopathogenic either maybe virus, bacteria, fungi are the most important for in future for the management. When we all know that the chemical is the we are doing the rush for that. So in the future, this is the most important thing. And for the organic point of view, as well as our Prime Minister doubling the farmer income, for that the organic agriculture is the most important. And in that way, this the entomopathogen is the play key role for the management of any insect. So thank you very much, and sorry continuously interpreting these things. So for the I am very thankful to for the organizing secretary for giving me chance to present this in the.
Thank you, Dr. D.M. Jetwa sir. So even though you are facing the problem of internet connectivity, and you have summarized very well your topic, and you have given a very nice information. So first of all, on behalf of organizing committee, I am highly thankful to you, Dr. D.M. Jetwa sir, for your kind consent to accept our invitation for delivering the guest lecture over here on the virtual platform. You have narrated so many things, and you have done a very good job at Junagadh Agriculture University in the field of the biopesticides, and you have narrated over here a very rare photos available, particularly for the biopesticides and other things, and you have supplied a large amount of the biopesticide to the farmers, and you have done it. We are proud of you, Dr. D.M. Jetwa sir, that you are here with us and give a very nice talk over here. So again, I am highly thankful to you, Dr. D.M. Jetwa sir. Now our next speaker, I request to Dr. Swapnil Deshmukh to invite the next speaker. This is the rapid fire round. Only 15 minutes given to the speakers, and within 15 minutes, I request you to conclude your topic.
Thank you, D.D. Patel sir. Now I invite Dr. Rishiketu Kumar sir to deliver his lecture. First of all, I will just give a short introduction about him. He has completed his doctorate degree from NDRI Karnal in the animal nutrition. Then he has more than 9 years of experience. He has published 12 research papers, 4 book chapters, and more than 35 popular articles. The main thing that I want to say here it is that he is not only a good professor, good scientist, but he is also a good artist. Because you can see that in our questionnaires we have put up one logo of our college. This logo has been created by Dr. P. Kumar sir. Okay, sir. Now the platform is yours, sir.
Okay, thank you, Dr. Deshmukh. First, I would like to thank Dr. D.D. Patel sir, who has given me such an opportunity to deliver lecture on this platform in this national level webinar. And as there is time limit and we are already running out of time, so I will not take much time in introduction and other things. So let's start. As my assigned topic is nutritional interventions to mitigate methane production in ruminants. And this topic is although different from other topics which we have gone through, because this is totally related with animal science, totally related with livestock production systems. And this topic is very important. Importance of this topic is not only for academicians and researchers, but this topic is equally important for farmers and livestock. And most importantly, this topic is important for environment and our mother earth. So we all know we are nowadays actually since 1 year facing COVID-19 problem, pandemic potential of COVID-19 we have seen. But this topic, as we are dealing with greenhouse gases, we are dealing with climate change. So this topic is having more pandemic potential than COVID-19. Only difference is that we are not here. It is not visible direct mortality, which we are seeing in COVID-19 cases, direct mortality and morbidity of human population. But here, abiotic as well as biotic, all components are suffering. due to climate change, proposition, even livestock, agriculture, and all other. So we all know about climate change and the gases responsible for climate change, mainly carbon dioxide, methane, nitrous oxide, sulfur dioxide, and out of these gases, most important is carbon dioxide and methane, but we will focus on methane in particular this topic. Why we will focus on methane? Because we are dealing with livestock in agricultural system, and livestock are main contributors to methane emission. So we will deal with methane. Again, important thing about methane is heat trapping potential. Heat trapping potential of methane is 21 times more than carbon dioxide. And total global warming contribution of livestock, mainly ruminants, in total global warming is around 15%. So this is very important topic, how to mitigate methane from ruminants, because their contribution is significant in climate change or greenhouse gases. Global warming and climate change is one of the most critical global challenges. We all know globally ruminants produce 80 million metric tons of methane manually. Out of this 80 million metric ton, livestock population from our country contributes around 11 million metric ton, means around 15 to 16% contribution is from our livestock. Reason is that we have large population of livestock. We have around 200 million of cattle, 109 million of buffaloes, 140 million of goat and sheep. So that's why we are a major contributor in greenhouse gases in terms of livestock through ruminants. In ruminants, 87% methane is produced in rumen and remaining 13% from hindgut fermentation. So mainly rumen is responsible for methane formation. Energy loss ranges from 2 to 12% of gross energy intake in cattle. One aspect of methane emission is that there is loss of energy, means loss of productivity in terms of methane, and that loss is 2 to 12%. So the necessity finding newer strategy to reduce methane production and enhance nutrient utilization. So if we are reducing or mitigating methane, it means simultaneously we are enhancing the production, because it is ultimately energy which we are losing in form of methane from our livestock. What is enteric methane? How it is formed, where it is formed, and what is the process of methanogenesis. As we can see, livestock ruminants mainly feed on carbohydrate in form of fodders, protein and fat. Their main diet consists of these three nutrients, and all these three nutrients get fermented inside the rumen and get fermented to products like short chain fatty acids. We also call them as volatile fatty acids. And these volatile fatty acids are mainly acetic acid, butyric acid, and propionic acid. In high roughage diet, acetate formation is more, around 60 to 70%, propionate 15 to 20%, and rest is butyrate. Simultaneously, with these VA phase, there is formation of hydrogen and carbon dioxide, and this hydrogen is being utilized by methanogens, different bacteria or archaeas. They used this hydrogen and carbon dioxide for synthesis of this methane, and finally methane is ejected from livestock through burping or belching. So that's the whole process of methanogenesis in the rumen. We can see here 87% methane is being synthesized in rumen, while 11% in large intestine, while 95 to 99% methane is ejected through burping action, and 1 to 5% is only through rectum, that is from dung. Enteric methane emission from ruminants, we can see cattle Contribution of cattle is around 77%, while contribution of buffalo is 14%, and small ruminants like sheep and goat, their contribution is around 9%. So mainly cattle is responsible for methane production because they are contributing significantly, and this is due to their large population all over the world. Because buffalo population is comparatively lesser in comparison to cattle and mainly restricted to Asian countries. Even in Asia, if we talk about buffalo population, around 57% of total world's buffalo is in our country itself. So it is somehow area specific, restricted to some Asian countries like India, Burma, Nepal, Pakistan. So their contribution is comparatively less. And methanogens are bacteria, rod-shaped or cocoid-shaped bacteria, which are responsible for methane production by utilizing hydrogen and carbon dioxide. Over 50 species of methanogens have been identified, but in case of rumen, some species like Methanobacterium formicum, Methanobacterium bryanti, Methanobrevibacter species, these are mainly responsible for methane synthesis, and they are responsible for 30 to 99% of methanogenesis inside rumen. While Methanomicrobium mobile and Methanocoelius species are responsible for 0 to 54%, range is very big, while Methanosarcina species, their contribution is around 2 to 3% as a whole in methanogenesis. Now the $1,000,000 question is, what is the need of methane mitigation? Why we are always talking about methane? And the reason I have already told, heat trapping capacity is 21 times more than carbon dioxide. And second one is half-life of methane is around 12 years. So it is not like that we will start today methane mitigating process or research and result will just start to come. So it will take time because half-life is more. So that's why we need to do this now. And what will be the advantage? Firstly, less methane means a lower concentration of greenhouse gases in the atmosphere. So climate change, we can somehow contribute in climate changes. We can control the greenhouse gases concentration. And secondly, less methane production or methane mitigation means increased efficiency of livestock production and increased income for farmers. As we have seen, 2 to 12% of gross energy loss is through methane in the form of methane. So that loss we can actually, animal can utilized in production of milk, meat and other products. So that will be actually a profit for farmers. This is a schematic diagram which shows these are unshaded areas and shaded areas. We can see unshaded areas are the targets and shaded areas are the options. So basically these are the option and these are the target unshaded area on which these options can act to control methane production inside rumen. For example, diets. We can change diet or we can make dietary modification like high concentrate diet, high digestible forage, balanced ration, fibrolytic enzymes. So they will these modification in diet will reduce methanogenesis in rumen. Similarly, on animal level, we can go for genetic selection. We can increase the productivity of animal. As a result, methane production per unit of product will decrease unproductive animal. It is not good to maintain unproductive animal in herd because that will be actually a loss for farmers and also loss for climate because they will contribute significantly in methane production, improve animal health and increase fertility. Nitrate and sulfate reducing bacteria, they act as a hydrogen sink and they get converted into nitrite and sulfite in that process. So they use hydrogen which is released in fermentation process, and so less hydrogen will be available for methanogens for synthesis of methane. As we can see here, they are acting on electron receptors, their target. So they can also mitigate the methanogenesis. Similarly, organic acid like malate, fumarate, and monensin, ionophore. Actually, monensin is ionophore like monensin, rumensin, lessalocid, selenomycin. So many ionophores are there which also decrease methane, because and their mode of action is actually different. They increase the feed conversion efficiency of animal and decrease the dry matter intake. So as a whole, methanogenesis will be there. They also alter the rumen fermentation pattern. Propionate will increase and acetate will decrease. So again, that will control the methanogenesis process, because acetate and butyrate synthesis promotes hydrogen generation, while propionate synthesis acts as sink for hydrogen, that is utilize hydrogen. We can use fumarate reducing bacteria. We can use secondary plant metabolites like saponins, tannins, as these are anti-protozoal in nature. Even tannin, mainly condensed tannins, are anti-protozoal in nature, and they also act as toxic to methanogens. Plus, they also hamper the fiber digestion. So less fiber digestion means less methane production in rumen. Similarly, some other options like halogenated compounds, plant metabolites, fatty acids, fats and oils, nitro compounds, bacteriocin, vaccine, we can all use this for mitigation of methane or in rumen. Acetate and butyrate promotes methane production. Here we can see in this reaction, acetate formation is generating hydrogen ion. 1 mole of acetate is generating actually 4 hydrogen. Similarly, here propionate is consuming hydrogen, that is electron receptor act as hydrogen sink, and butyrate is also generating hydrogen. So formation of acetate and butyrate is hydrogen generating, while formation of or synthesis of propionate is hydrogen consuming. That is, propionate synthesis acts as a sink of hydrogen, and so one of the way for mitigation of methane inside the rumen. These are the various strategies which we use for methane mitigation inside rumen. These are nutritional strategies. By using carbohydrates, frequency of feeding, we can change the frequency of feeding. Forage species and maturity, its maturity also affect methanogenesis. Processing of forage, silage feeding, complete feed block or TMR, that is total mixed ration. Many a time we offer animal individual feed. First we offer dry straw, after then at the time of milking concentrate and green fodder like that. But better we should offer in the form of TMR, mix the whole the feed together, dry fodder, green fodder and concentrate, and offer in that way. So that will actually alter the rumen fermentation, that will promote the synthesis of propionate, and as a result methane mitigation will be there. Manipulation of rumen fermentation we can do by using these ionophores. Defaunation. Defaunation means removal of protozoa. We use anti-protozoal agents. Even plant secondary metabolites like saponin and tannin are also anti-protozoal agents. So removal of protozoa will result in less methane production, because methanogens or methane synthesizing bacteria are attached with protozoa in a symbiotic relationship with protozoa. So if protozoal population will be less, methane production will be less. Addition of fat and oil. Addition of fat and oil is also essential, because if we use unsaturated fats in animal feed, the hydrogen ion generated in fermentation will be utilized by those unsaturated fatty acids, and as a result less methanogenesis will be there. Plus excess of fat also hamper the fiber digestion. As a result less methanogenesis, because less acetate formation. We use probiotics. We all are aware of probiotics like yeast, Saccharomyces, Lactobacillus. There are so many probiotic organisms which alter the ruminal fermentation, which increase feed conversion efficiency and reduce the methane mitigation. Similarly, we use plant secondary compounds, immunization, use of chemicals. We can promote the reductive acetogenesis process in rumen. We use probiotics, rumen methane oxidizer, bacteriocins, and bacteriophages. All these will manipulate the rumen fermentation, and as a result, there will be less methane mitigation. Secondly, we can go for managemental strategies like number and productivity of animals. We can decrease the unproductive animals in our herd. That is important. And if we can increase the productivity of animals, then methane production per unit product will be decreased. Genetic selection of animal based on their residual feed intake capacity. So all these nutritional strategy as well as managemental strategies are very useful in mitigating methane production in ruminants. Here we can see improved forage quality and management. Digestibility will increase. As a result, methane reduction will be increased. Similarly, mechanical feed processing also increases forage digestibility, less retention time in rumen, so methanogen will get less time to act on. As a result, reduction in methane production. Feeding concentrate diet will hamper the acetate formation and promote the propionate formation. So there will be less methane production. Also, on high concentrate diet, ruminal pH goes down, and at low ruminal pH, there will be less methanogenesis. But problem with concentrate feeding, if we are feeding concentrate more than 50% only, then this methane mitigation is visible. Otherwise not. And even more than 80 to, if we are using 80 to 90% of concentrate in diet, there is significantly less methane production in rumen. But in our condition, in Indian condition, it is not possible to use 80 to 90% of concentrate due to low producing animal and not a good economic condition of our farmers. Nutrient balancing program will also result in decreased methane production, but these all feeds and feeding practices are only applicable. These can be applicable in all systems, but nutrient balancing and precision feeding is applicable in only confined system, not in grazing system or pasture-based system. Frequency of feeding, low frequency feeding, increased propionate and reduced acetic acid production, as a result lowered methane production. Because low frequency feeding result in increased diurnal fluctuations in ruminal pH, which is inhibitory to methanogens. Forage species and its stage of growth. For example, leguminous forages. On leguminous forages, methane production will be less in compared to cereal forages due to high digestibility. Processing of forage, grinding or pelleting results in decreasing methane losses up to 20 to 40%. Reason is lower fiber digestibility, decreased ruminal time available for organic matter and fermentation, and faster rate of passage. If we grind the fodder, chop the fodder, then digestibility of fiber decreases due to their faster passage rate from the rumen. So microbes get lesser time to act on. As a result, less acetate formation and less methane formation. Silage feeding also, ruminal fermentation of silage results in higher butyrate and lower acetate production, which helps in mitigation of methane or less methanogenesis. Similarly, complete feed block or total mixed ration we have discussed in that table. Methane production decreases with increasing concentrate proportion of diets, replacing fibrous concentrates with starchy concentrates, increasing the digestibility of forage with legume compared to grass forages, and with silage compared to hay. Hay is actually many a time this is synonym to a straw, and a straw are rich in fiber. So If animal will feed on hay or a straw, there will be more acetate production so there will be more methane production due to more release of hydrogen ion. Feed supplements and feed additives. If we use ionophores, we have discussed like monensin, lessalocid, they will act as hydrogen sink. They will promote the synthesis of propionate. As a result, methane production will be less. But they will impact the productivity. And their limitation is microbial adaptation. After a certain time, microbes will get adapted to ionophores, even organic acids, plant extracts, halogenated compounds, and then we will not use to see that significant results in methane production. Organic acids, nitrate, malate, fumarate also acts as hydrogen sink. Plant extracts we have discussed in previous table like tannin and saponin acts as anti-protozoal agent as well as hydrogen sink. Even tannin is toxic to some methanogens. They also decrease fiber digestion. So as a result, there will be less methane production. Dietary lipids we have discussed. Halogenated compounds like bromochloroethane, chloroform, also toxic to methanogens, and demerit is they also decrease dry matter intake. So as a result, there will be lesser methane production. Monensin and is very important ionophore, which results in a reduction of gram-positive bacteria, concurrent shift in fermentation from acetate to propionate, and that result in decreasing methane production inside rumen. Electron acceptors, we use nitrate and sulfate, which get reduced to nitrite and sulfite in rumen by using hydrogen ion, which is emitted during fermentation process, and as a result there will be less hydrogen ion available for methanogens and methane production will decrease. Even we can use nitrate as a nitrogen supplement to low quality crop residue based diets as a NPN source, non-protein nitrogen source. Bacteriocins are another important aspect, are the proteins produced by bacteria that we can use for methane as a methane mitigating agents. For example, one of the important bacteriocin is nisin produced by Lactobacillus lactis strain, which stimulate propionate production and reduce methanogenesis by 36% in vitro. But this is under in vitro condition, not in vivo condition. And the reason is that bacteriocins also inhibit methanogens population in the rumen as per Sir and co-worker 2005. Reductive acetogenesis process is another process which acts as hydrogen sink because we use to isolate acetogens from rumen and again use to inoculate them in rumen after growing and culturing them in lab. And what they used to do, they used to synthesize acetate from hydrogen ion and carbon dioxide. We can see here acetogens use hydrogen ion and carbon dioxide for synthesis of acetic acid, and in that process they use hydrogen, acts as hydrogen sink. As a result, methane production will be decreased. Immunization, even some scientists in Australia have developed a vaccine and they have patented that vaccine for methane mitigation, and they have used this vaccine also. So we can immunize animals against their own methanogens and protozoa, and we can prepare these vaccines from the methanogens isolated from animal itself, and again we use for the prevention or mitigation of those methanogens. Immunization of sheep has been done with a mixed wholesale preparation from 3 methanogens and have seen that reduced methane production by 7.7%.
Dr. Sir, it is now high time, sir.
Just 2 to 3 minutes. Okay. Genetic selection of animal based on residual feed intake. Residual feed intake concept is very good concept, and it is one of the long lasting method for methane production we can use for future. RFI residual feed intake means this is difference between predicted feed intake and actual feed intake. What we are predicting for animal and what animal are eating. That difference is residual feed intake. So animals with low RFI are considered good because there will be no negative impact on their productivity. So we can select animal with low RFI, and that will result in less dry matter intake. As a result, there will be less methane production. So as a long term, we can go for this. Number and productivity of animals, as we have already discussed, the culling of non-productive and low-producing animal can result in methane mitigation. Increasing the productivity of animals can also result in methane mitigation per unit of products. Finally, we can conclude a strategy like increased grain feeding, forage processing, and pelleting, organic acid supplementation. These are not suitable for low-cost products. Whales and defaunation have remarkable ability to reduce methane emission, but problem is fiber digestibility because they will hamper the fiber digestibility significantly. Addition of ionophores to ruminant diet offers opportunity to reduce methane production, but rumen microbes can adapt to them. So adaptability is one of the problem. Technology such as plant secondary metabolites, a stimulation of acetogens, and immunization against methanogens have emerged to lower methane production. Again, genetic selection, as we have discussed in RFI concept. So genetic selection is the area of research with the best chance of finding a solution, but it is expensive and long-term process. Vaccination and probiotics are promising approaches for future research without any hazard to animal or environment. Supplementation of diets with ionophores concentrates and urea molasses mineral block could be cost effective for high yielding animals in our situation, in Indian situation. Now, concluding remarks, mitigation of methane emission can be effectively achieved by strategies that are cost effective, improve feed conversion efficiency, improve animal productivity, and have no potential negative effects on livestock production, hold a greater chance of being adopted by producers. So ultimately, whatever mitigation strategy we can adopt or we will adopt, that should be cost effective. There should be no any negative impact on animal performance, no any negative impact on feed conversion efficiency, and that will be better. Thank you all of you. If any query, please.
Thank you, Prishketu Kumar sir. It was a very nice lecture. We will see you for the chat box. It was a very nice lecture. You have covered everything about nutritional interventions that can mitigate our this methane emission, and surely participants have been benefited by your presentation. Thank you very much, sir. Now we'll invite our next speaker, Dr. H.H. Patel, sir. He is my senior in the Department of Agronomy, and he has experience of more than 6 years of experience, and he is a very eminent scientist and a very good professor. Now he will be delivering the talk on enhancing water use efficiency. It is a very important topic. So before starting his lecture, I have an announcement that after tomorrow's lecture, it is very good lecture tomorrow morning, and after the lecture at 11 a.m. there will be the validity function. So you all are requested to join that validity function. And okay, so I request Dr. H.H. Patel sir to start his lecture. H. Patel, sir. Now the platform is yours.
Okay, Dr. Swapnil, I am audible?
Yes, sir. You are audible.
Okay, thank you, Dr. Swapnil, for nice and impressive introduction. I am very much aware that this is a time of lunch. Even though I observed that more than 70 participants over here, it's really appreciated. I am sure that distinguished participants are very much learned persons of the topic. However, I am trying to say this important point regarding my topic, enhancing water use efficiency need of an hour.
Sir, please share your screen.
Yeah, I am sharing. Okay, my screen is visible?
Yes, sir. It is visible.
Okay, thank you. We all know that Earth around 70% of geographical area is covered by water body. So we think that we have enough plenty of water. However, only 3% of fresh water is available for human purpose, like domestic, industrial, as well as agriculture purpose. Among these available water, fresh available water, 2/3 of fresh available water is not available. So as it's stored in form of glacier, while only 1/3 of fresh water is available for their use. All over the world, 1.1 billion people faced difficulty to access the water, and 2.7 billion people face water scarce for at least 1 month in a year. Besides, approximately 2.4 billion people did not find good quality water for drinking purpose, have to compromise with health issue like cholera, typhoid, and other waterborne illnesses. From diarrheal disease, many of the water systems that keep ecosystem flourish and feed a growing human population have become stressed. Most of the river, lakes, and other aquifers are drying up or becoming too pollutant, hence not suitable for its use. Then presently, as per report, more than half of the world wetland have been disappeared. So agriculture require more water than any other sources and waste much of the water due to insufficient irrigation technique. Because in India, more than 80% of irrigation applied to the surface matter, which causes waterlogging problems and find low water use efficiency. Along with this, climate change is altering the weather patterns and water around the world, causing a water shortage and drought in some area or flood in another. At present consumption rate, this situation gets worse. By 20 to 2050, 2/3 of the world population may faces water shortage, and ecosystem around the world will suffer even more worse. So enhancing water use efficiency to different technique is need of today. If we want to enhancing the water use efficiency, so we want to increase the output or productivity of the water. Otherwise, we want to reduce the consumptive use of water. The global water budget is around annually 86 centimeter annual precipitation over the globe. Out of this, 77% of precipitations fall over on ocean and 23% on land. Out of this 23% precipitation, 16% of total precipitation is received losses through evapotranspiration from land, and only 7% of the total precipitation received by the land uh, return to the sea as river and ground water flow. So in India's water budget, India receives 3,838 kilometer cube precipitation. Out of this, potential flow in river is 1,869 kilometer cube, and natural recharge is 432 kilometer cube. So India having the available water is 2,300 kilometer cube, and remaining 1,537 kilometer cube losses through evapotranspirations. Now, in India, the per capita water availability is in the year of 1961, it is too high, for 4,467 meter cube per annum per capita. But nowadays, in 2015, it is reduced up to 1,567 meter cube per annum. And up to 20 to 25, the population of India is increasing day by day. So per capita water availability is decreasing. In some areas of Gujarat and Rajasthan, in northern Gujarat or in Saurashtra or in Rajasthan, the chronic water scarcity is available, because there are less than 1,000 meter cube per annum uh, water is there. Water demand in India. In the year 2010, the irrigations required 550 billion cubic meter water, and for 42.5 billion cubic meter for domestic purpose, 37 billion cubic meter for industrial purpose, 18.5 billion cubic meter for power productions. While in the year 2050, the estimated water demand in India is 107.5 billion cubic meter for irrigation, more than 100 billion cubic meter for domestic purpose, more than 80 billion cubic meter for industrial purpose, and more than 70 million cubic meter for power purpose. Means the requirement or the demands of water is increasing day by day. The domestic and industrial demand of water is growing faster than irrigation needs, because in power production there is a 4 times higher within 40 years. We required large number of water. This is the land statistic of India. India having a 329 million geographical area. Out of this, 141 million hectare net crop area and 195 million gross crop area. Out of this, 40% area is irrigated area, while 60% area is rain-fed area. This is crop by irrigation coverage. Sugarcane in case of irrigations, the sugarcane reaches 94% area of sugarcane are irrigated, while in soybean only 1% soybean area is irrigated. So we are from this slide we are stated that if we are increasing the water use efficiency, then high demanding crop are high water demanding crop are reduced, and the low demanding crops are increased day by day. Now this is the question: what should be irrigated, land or crop? Farmers are just irrigate the land, not the crop. If we are wasting the water to irrigate the land, then the water use efficiency is decreases. But when we are applying the drip irrigation methods, so we are each and every drop of water is utilized by the crop and increase the water use efficiency. These are the some efficiency of irrigation systems. The conveyance efficiency of surface irrigation is too low. Only 40% of water reaches from source to field. While in application efficiency, we are not much aware about this application, because we are applying surface irrigation method. So efficiency of application efficiency of surface irrigation water is only 60%. While in sprinkler it is reaches up to 70%, and highest application efficiency found in drip, 90%. So we are applying water through drip is increase the water use efficiency. If we want to increase the water use efficiency, that increase the yield or reducing the consumptive use of water. So in evapotranspiration cases, surface and sprinkler required surface and sprinkler method of irrigation losses the 30% of irrigation water through evapotranspiration, while in drip irrigation method only 20% water is evaporated. Overall efficiency is too low in surface irrigation method, while drip irrigation method having the highest overall irrigation efficiency, 80%. So this is the micro irrigation system. Drip irrigation system having save the water up to 50 to 70% and increase the crop production up to 20 to 70%. While in sprinkler irrigation system, save the water up to 30 to 60% and crop production increase up to 20 to 50%. In drip irrigation system, we are using water drop by drop near the root zone area. So we are not flood or do not irrigate the land, only we are irrigate the crop. So irrigation efficiency increase through drip irrigation systems. Nutrient distribution pattern. If we are applying nutrient in surface irrigation methods, so nutrient leak down so like this, and we are applying fertilizer in drip, so it is a localized placement, but we are applying fertilize with drip fertigation, so uniform distribution of fertilizers, nutrients, uniform placement of fertilizers taken place. So drip fertigation is the best method to apply nutrients. Why efficiency of surface irrigation is low? Because in surface irrigation water we are applying water haphazardly, uneven field surface and uneven slope of the field. Next one is evaporation from field, because we are applying the water to field, not to the crop. We are not applying evaporation from field channels and network. We all know that our network channel canal network are very poor, and it is urban or open channels, so it is evaporated through network channels, losses from field channels, and water logging during flood watering. We are applying high amount of water in surface irrigation, so even though the slope of the land is not correct in our field, so we are not farmers are not leveling their field regularly, so water logging is the major problem. So during the from the water logging, the production is decreases, so water use efficiency is always decreases from the water logging. How to improve water efficiency? First is soil leveling. So we are slow, level the field, or we are sloped up to 2%. So it increase the it is decrease the water use, and decreases the time of water applications. So it increase the water use efficiency. Then method of irrigation. If we are applying irrigation through drip irrigations or sprinkler irrigation, it increase the water use efficiency instead of surface irrigation. If we are applying the surface irrigation through ridges and furrow, means in control method, that it increase the water use efficiency instead of uncontrolled methods. Scheduling of irrigation, planting techniques like pair row planting or broadbed and furrow. These planting techniques are also useful for increasing the water use efficiency. Scheduling of irrigation, like irrigate the crop at their critical growth stages, or at IWCP approaches, or like canopy temperature. So it increase the water use efficiency. Mulching, the best method to increase the water use efficiency is mulching, because we cover the soil with artificial plastic or organic materials like paddy straw, sugarcane straw, or sugarcane species, or wheat busas. So it conserves the soil moisture and reduce the evaporation losses, and also reduce the weed problem. Uh, if we are controlling the weeds, then it reduce the transpiration losses. So it increase the water use efficiency, use of anti-transpirants, in those, these are the anti-transpirant used to close reduce the opening time of stomata, and reduce the transpiration losses. This is my PhD experiment. We are in my experiment, effect of planting techniques and irrigation level on chickpea. So I found that we are applying for three plots, three treatments of irrigations, and 4 treatment of planting techniques. We are applying 1 irrigation at branching, 1 irrigation and pod initiation, and third one is 2 irrigation at branching and pod initiation. And in subplots, in planting techniques, we are sow the crop at flat bed sowing, furrow after 2 row, furrow after 3 row, and furrow after 4 row. And with this, we are without irrigations and flat bed sowing. So we are here, we can show here that the control means there is no irrigations and no planting techniques is used. So grain yield of the chickpea is only 896. But here the treatment means all the irrigations techniques, irrigation methods, and planting techniques average is 156. Means we are increasing the yield by giving only 1 irrigations or only 2 irrigations with the help of planting techniques like furrow after 2 or 3 or 4 rows. Uh, Here we are reducing the consumptive use by applying 1 irrigations at the branching stage. So we are increasing water use efficiency. If we increase the we are apply 1 irrigation at branching stage instead of 2 irrigation at branching and pod initiation stage, we are increasing the water use efficiency, and we are also increasing the nearly 100 kg more yield than the control. Effect of laser lane leveling on wheat. This experiment conducted at Navsari by Dr. Das in 2018. So here he had taken 3 treatment for land leveling, 0.05% slope with 0.15% slope and 0.3% slope. They found that they increased the grain yield in 0.15% slope, 3,475 kg. Means if we are level the field at the 0.15% gradient, so it increased the water grain yield and water saving up to with 565 meter cube per hectare, and water productivity is also increased by 1.12% kg grain per meter cube water. Patel et al. Conducted experiment at SDAU Gujarat. They tested 3 irrigation practices, 0.6 IWCP ratio, 0.8 IWCP ratio, and 1 IWCP ratio with different mulches like mustard straw, castor shell, 5 ton per hectare, and soil mulch. They found that one IWCP ratio increase the grain yield, and the castor shell and mustard straw mulches also increase the grain yield and fodder yield, and increase the water use efficiency, and also increase the net realization and BCRs. Savani et al. Conducted an experiment at Soil and Water Soil and Water Management Research Unit at Navsari Agriculture University in Navsari. They tested drip irrigation. They tested different level of irrigation and mulch on Rabi PGNP. So they found that applications of irrigation at 0.8 pan evaporation fractions increase the grain yield, and water saves by and at 0.6 pan evaporation fractions, grain yield also increased, and water saving by 32%, and water use efficiency is also increasing. And black plastic mulch also increase the grain yield, and sugarcane traces is also increase the grain yield. Then no mulch. So it is highly if we are using we are increasing the water use efficiency, means we are increasing the yield by the mulch, so it increase the water use efficiency. Effect of different anti-transpirant on yield of castor. Patel conducted experiment at SK Nagar. They uses kaolinite, PMA, cetyl alcohol, water spray and control. They find kaolin as an anti-transpirant, increasing the yield and moisture percentage at harvest at 2.23%. So kaolin reduce the stomatal opening and reduce the transpiration losses. So it is increase the water use efficiency. Now I am concluding this topic. Irrigation remain vital importance in India and worldwide as a means to enhance production and water use efficiency, because water is the nowadays water is the limiting factor. Many organic engineering and management technology like agronomic practices like planting techniques, irrigation methods, engineering like leveling, and other management practices like mulches can reduce non-productive use of irrigation water, because we are mulch the field or we are level the field, then it reduce the water use by the crop. However, in some cases, increasing irrigation efficiency may not simply achieve new water for allocation unless the consumptive use part of diverted water is actually reduced. In that reference, it is summarized this opportunity as increasing output per unit of evapotranspiration, reduce losses of usable water to sink, reducing water from pollutant, from sediments, nutrients, and other agrochemicals, and reallocating water from lower value to higher value use. Means we are applying uh, irrigation to sugarcane, then we are applying water to soybean. It increases the water use efficiency, because in paddy, in sugarcane, we are required large amount of water, while in soybean or in pulses, we are irrigate the crop at least more only one or two irrigations. So it increases the water productivity and water use efficiency. So thank you for be with us. Save water is start with you. So we are increasing the water use efficiency of nations, of world, of our state. Okay, thank you, Dr. Swapnil.
Okay, thank you, Dr. H.H. Patel, sir. Actually, it is very important topic related to sustainable agriculture, as water is always a limiting factor for our agriculture. So you have delivered everything that is beneficial for water use efficiency. You have shared your practical experiences with us, and I am sure that our participants will be benefited with this. So thank you very much, sir. Now we will conclude our today's this webinar, and tomorrow we will be meeting at 10 a.m. morning, and there is one very eminent speaker, Dr. Matt Newland. He is a foreign speaker with us tomorrow. So please, I request all the participants to be remain present for there for the lecture. and after lecture that will be the validity this session. And before concluding, I will be very much I'm thankful to Dr. Rihanna Habib Khan, Professor and Head, Scout Kashmir. Your positive response is always being very appreciated by us. Thank you very much. So we will end the